Okay, welcome to the morning session on anthropogenic effects. I am Andrzej Rakowski, and I will guide you through the morning session as smooth as possible. And we have 6 talks. The first talk will be the tribute to our beloved colleague Ingeborg Levin, who passed away in February this year. And I think most of the people in this room knows her, if not personally, met her on some conference, and we know how important for us, and especially for this conference, which is radiocarbon in the environment. So in this session, all of the presentation probably use the data set from Schau Insland and Jungfrau Joch, and her really dedicated work on this. We have to remember her and the contribution in the science she put. So on the end of the presentation, I will ask you, instead of question and answer, maybe we can give her a minute of silence to memorize her, to memorize the last time we met her, if you met her. I remember, for me, it was a conference in Paris in 2012. She was there, vigorous, as always, presenting all the results she achieved. So, okay, we can start. So Samuel will present us the legacy, scientific legacy of her.
Good morning, everybody. My name is Samuel Hammer, and I had the pleasure to work with Ingeborg for the last 20 years, and together we set up the ICOS Central Radiocarbon Laboratory. And when Mikhail wrote me an email that he had this idea that at this conference there should be a tribute to Ingeborg, I was thinking that that was a very good idea, and I happily came here to give you this talk about Ingeborg, about her accomplishments in the past. And from time to time I will sneak in a little bit of our actual research. So where are we currently? What was the past? Where was it beginning with Ingeborg, and how does it develop? So the authors is me and the entire radiocarbon team of the Central Radiocarbon Laboratory. So if we think about Ingeborg's legacy, the first thing which may come into your mind is the wealth of atmospheric radiocarbon data which she produced over her years. And therefore I put one of Ingeborg's notebooks here. It's actually her first lab book which she had. It says Laborbuch in German, Ingeborg Levin. And over the course of her life, she filled very many of those lab books, and we can just take a look inside. And what you see up there is actually the first sample of radiocarbon which she took in Heidelberg. It was in October 1976, integrated over 2 weeks, and the atmosphere in Heidelberg at that time had an activity of 312 per mil. To the right, you see a picture of Ingeborg just taken in the last fall, when she was standing at the castle of Heidelberg, looking over the city, and you see until the end of her life she was at good health. She was always working quite a lot, still 8 hours a day. She was very lively, and it was just really a drop out of the full life down to nothing. Yeah? I think she would have wanted it like this. So yeah, you can see it like that. When I was a young student, I argued with Ingeborg, why do you put all the different measurements in your notebooks by hand? Don't you have more important things to do? And then she told me, no, this is really important, because I only do this at the end of the day when my brain is not working well anymore and I cannot be creative. But then I still have sufficient time to just write down those numbers. And when I write down those numbers, it gives me the feeling of the laboratory and the quality which the laboratory is currently producing. How well do the measurements perform? And by just doing this and writing it from hand, I get the feeling directly. The second reason for her was when she did that for all the different stations which she had over the globe, she had basically her hand on the pulse of the radiocarbon concentrations, and you could see them going down and their changes which they had over the years. So for her it was a very good way of actually staying in contact with her data. There are many ways to give this talk and to look back on Ingeborg's scientific career. I decided to start with her Diplomarbeit, which is nowadays a master thesis, and we want to look into that, what she already achieved there. So the thesis was titled Regional Modeling of Atmospheric CO2 based on C13 and C14 measurements. And she handed that in 1978. Sorry, wrong direction. So let's go to the content of this. So first of all, she established 2 background stations in that thesis. That is Jungfraujoch and Schauinsland, where she started the measurements, and she introduced this so-called Raschig tube sampler. It is a sampler which contains sodium hydroxide solution, and you can bubble air through it over the course of 2 weeks. In total, we use 25 cubic meters of air, and then you absorb the CO2 from the air in the sodium hydroxide solution, ship it back to our laboratory, where we can then extract it and measure it for radiocarbon. That system is still nowadays running at all the ICOS stations. It's a little bit clumsy, not very small and elegant, but it's really a workhorse, and it did great work over the last 50 years. Here you see Ingeborg when she was installing the first sampler at Schauinsland station, and you can, maybe some of you in the front can see at the bottom, this is the Raschig tube, which is turning while we are pumping the air over it. So she installed those background measurements, and at the same time she installed measurements at Heidelberg, also for radiocarbon. And then she looked, what can I say about the excess CO2 concentration? So excess CO2 concentration means CO2 concentration above a background CO2 value. How is that composed out of biogenic and of fossil sources? And that is what you can see in that graph from her thesis. If the total excess ratio would be fossil, then you would follow this line down here. If it would be biogenic, it would be straight parallel to the x-axis. And from already that first plot, you can see that in wintertime, these are the field symbols, the excess is mainly from fossil nature, while in summer respiration is a dominant source for the CO2 excess, even in a city like Heidelberg. So she did not only look into radiocarbon in CO2, but she also made first measurements of radiocarbon in methane. By that time, I think that was quite early to do that, in the 78 or 76. And here we have a similar plot where we are looking on the x-axis at the methane concentration and on the y-axis the radiocarbon content in methane. And if also this methane excess would be of fossil origin, then the measurements should follow this lower line, which they clearly don't. So from this plot, it was clear that the methane enhancement is not dominated by fossil fuels. It's dominated by biogenic sources. The thesis was titled Modeling, so she developed and programmed a Gaussian plume model in Fortran, where she was modeling CO2 enhancement, fossil fuel CO2 enhancement, and she compared them to the measurements which she performed in Heidelberg, and you can see the correlation plots of these two quantities. And still nowadays, for comparison between observations and models, it's not so bad. The next topic which she tackled in her thesis was the estimation of nuclear radiocarbon contamination from power plants. A problem we are still facing today, and we are still suffering from it. So she did that for radiocarbon in methane, radiocarbon in CO2, and she did it via direct air sampling or leaf samples. And all of that in 54 pages and about 2 years. So if I look back at that thesis, I'm questioning myself, so what are we doing? So we are still working on exactly the same stuff that she has started so many years ago. So yeah, it's really astonishing for me to see what she already accomplished back then. Okay, so that was the start of 48 years of passionate scientific work, and I would like to put her life, or her scientific life, on that time axis. So we just now started in 1976, and you see those topics, and we will go up to the recent past. In the top bar of that timeline, you will see everything which has to do with radiocarbon, fossil fuels, anything which led to the development of ICOS. And in the lower part, you will see all of the topics which she worked with, which were not related to CO2 or radiocarbon, like other greenhouse gases and stuff. And I will not be able to talk about the lower part at all. Okay, so the next thing, in the 80s, she started to set up her radiocarbon global cooperative air sampling network. And in the 80s, it was Neumeier in the Antarctica, which was the next station which was set up. And over the course of the year, this map developed, and you all might be familiar with those names from those stations because you might have used these measurements from Ingeborg. And for her, it was very important that she had a network which had a global coverage and which had continuous observations and was not interrupted. And by that time, when she started it, that was really a challenge because she had to go from a 3-year project to the next, and nothing was foreseen for continuous monitoring. So she basically had to make sure that sometimes she took a little bit of money from here to fund those measurements, sometimes from there. Then we only kept on sampling. We didn't have money for the analysis, but we stored the samples, and then when money was coming back, you try to do the analysis to a later point in time. Yeah? But for her, it was always clear that these data will be very, very valuable for the community, and that's why she kept on to run and operate these networks. But she also was interested herself in those observations, and she used them to understand the carbon cycle. And like any good environmental physicist, she loved to do that with low-dimensional models, which you can really understand and work with them. So what we are seeing here is a representation of the Grace box model, which was then finally in that stage in 2005 when she worked together with Tobias Nägler. What you have here is an atmosphere divided into 30-degree latitudinal bins from south to the north, and you have a vertical layering for the troposphere in 9 kilometers, and then you have 3 layers in the stratosphere, and that was very important in order to get the production mechanisms for C14 and the vertical distributions of them right. So with that atmosphere, she drove it with the input for bomb radiocarbon, radiocarbon from the nuclear industry, and natural radiocarbon. Then we had fossil fuels as another contributor for the CO2 and the land use change. And that atmosphere was coupled to the biosphere model, which contained of a 3-pool biosphere, which was leaves, twigs, and wood, and the soil, which was not very relevant on those timescale where the model was operating, and then to an ocean. And with that simple representation of the major parts of the carbon cycle, she was then able to really use the radiocarbon data, their distribution in space and also vertically, to say something about the major cross fluxes of carbon between those large reservoirs. And she did that by exploiting the information which was contained in the radiocarbon bomb signal. So what you're seeing here on the x-axis is time, and on the y-axis it's the bomb radiocarbon inventory, so the amount of radiocarbon atoms additionally introduced by the nuclear testings. So in this model, she basically used the Grace model to distribute this bomb radiocarbon between the main reservoirs on Earth, and you see in yellow or in orange, that's the troposphere. In red you have stratosphere, blue ocean, and then the biosphere in green, which was treated as a residual between the other two. And that data was really for her the point that she could reduce the cross-exchange fluxes between atmosphere and ocean from about 90 petagrams to 75. So that was really a major change in the knowledge of how those great or how those large CO2 reservoirs exchange with each other. And that was the beauty that one can use radiocarbon to do that. Okay, so where are we now? Today we are still operating most of those stations from the global network which Ingeborg established, but luckily today there are more players who do global background radiocarbon observations, and on that map you see the 4 groups which do mainly, or which have background networks for radiocarbon observations. It's Heidelberg and ICOS in black, those were the stations you have seen. Then we have NOAA, INSTA in blue, and in green you have Bering Head, New Zealand, and the red dots, these are the stations from Scripps, who do that together with the University of San Diego. And you now see also the different time steps on those stations, which tell you for which time periods the data are available. And in a European project which we're currently running, we wanted to put together all of those global background data set for our modeling colleagues in order that they can use those as background information. But if you want to do that, the question arises quite quickly, how well are those global networks compatible? And can we just use all of those in the same data set and combine them, or are we introducing systematic biases due to the different networks and the way they are working? So to answer that, we just conducted the second round of a pure CO2 radiocarbon laboratory intercomparison. Some of you in the room participated in the first round, that was 2016, a couple of years ago, when we distributed small ampoules of CO2. To do that, we just repeated this exercise, but now with a lot of more samples. And what you're seeing here is basically how we are preparing those samples. We have quite a large volume of CO2, which we get from the sodium hydroxide solution samples, 8.5 liters, and we distribute them into aliquots of 0.3 milliliters. And those of you in front might see the photo in the front, so it's just a flame seal CO2 aliquot, which we then distribute. For that exercise, in total, we prepared about 300 aliquots, and we distributed around 50 to those 4 laboratories, and that is then how this looks like while we were producing all of those brake seals in our laboratory and preparing them for shipment to the laboratories. I don't have now the time to go into all the details of the results, and it's also still an ongoing work because we are missing the final results from UCI for a couple of samples which have not yet been measured. But what you see here is the intermediate evaluation of this exercise for the ambient radiocarbon activity range. And what you see is that those 4 labs which operate the 4 networks do agree within a level of 0.5 per mil. And that is something which we are really extremely happy to see. That's what we always wanted to achieve, because the change of the interhemispheric gradient in radiocarbon north to south is about 0.5 per mil per year. Yeah, so if you want to be able to resolve this large-scale signal, you need to be sure that your networks are compatible on such a level. And we're quite happy that we have reached that so far, and we will continue that work once the final data are there and then publish it. But we have now very good confidence that at least for this point in time, these networks are compatible. It's hard to say what was 10 years ago, yeah? Were they 10 years ago compatible as well? But if you have a rigorous quality control in your own lab and you already had that 10 years ago, you might be able to extrapolate backward in time and say that it was okay-ish back then. We also had the first round in 2016, which was okay, but we could not go down to a level of 0.5 per mil because just we did not have sufficient samples to do the statistics to go down to this level. But within the uncertainties also back then, we thought the networks would agree. So as I said, there is this European project Corso, which which gathered all of those data. And okay, it's a pity, but that QR code behind the plant, if you would scan it, it would lead you directly to the report which we have written. That's a deliverable. You can just go on the webpage and look up Corso project deliverables, and then you will find deliverable 3.1, where you have the report where we compared all of those different networks, and in that report you will find a link to download all of those data. So these are free to use from all of those laboratories, and I have to say I am extremely happy that we did that last fall, because that was when Ingeborg was still around and we decided that we make all of her data publicly available. If I imagine that she would have passed away and we wouldn't have done that, then I would now be sitting there with that wealth of data, and I would have to decide, is it okay to give it out, or what do I have to do first, and would it be her will? No, like this, it's perfect. Yeah? We did it together, and for all of you, just go out there, use the data. Corso deliverable 3.1 if you want to look it up in Google. Okay, so far about the global work. Ingeborg also worked on the local and regional scales, and when she did that, the focus was fossil fuel determination. And so I would now spend a couple of minutes for her work on fossil fuels. So that is the well-known bump spike, and what you see in the lower part is a zoom to the last 35 years, where you have in blue and green the background curves from Schauinsland, or a marine background, in that case it's Macehead, and in red you see the observations which we perform in Heidelberg, and the depletions which you see in the atmosphere in Heidelberg can then directly be converted into a fossil fuel concentration, and if you measure the total concentration offset at the same time, offset with respect to a background, then you can partition this offset into the fossil and the biogenic, or let's call it non-fossil, component. And this partitioning between fossil and biogenic CO2 signals was really the argument which convinced the stakeholders that radiocarbon should become one of the key variables in the ICOS network. ICOS, for some of you, you might know it very well. For others, it's the Integrated Carbon Observation System. It's a European research infrastructure which aims for delivering high-precision observational data for the carbon cycle. That means we do atmospheric measurements, but at the same time we do measurements of PCO2 in the ocean. We measure fluxes of in-eddy covariant systems over the ecosystems. So it's really an infrastructure which encompasses all the major compartments of the carbon cycle. And the fact that we are now having ICOS is really something where Ingeborg was lobbying for 20 years for, ringing bells and talking to many stakeholders and convincing them that such a research infrastructure will be very needed for the European research field. And she was successful, and together with others, she managed to establish it. And what you are seeing on that map is the current European ICOS radiocarbon network, which we are having. So for all of those stations, ranging from very north Zeppelin, now down to Lampedusa in Italy, we have the measurements of radiocarbon available. So all of those stations have integrated 2-week sampling of radiocarbon, and in addition, all of those stations have a flask sampler, and a flask sample is collected basically over the course of 1 hour. You collect 6 liters of air, which are then analyzed for all of the greenhouse gases, and eventually also radiocarbon. It's nice, we're having those 21 stations, although if you look at the spatial distribution, you would say, weird, why did you distribute your stations like this? The answer to that is it's not us who are distributing the stations, it's the nations who are committing into ICOS and who are bringing forward their stations to become part of ICOS. And unfortunately, we have not been you stopped me. We have not been successful in convincing France and Portugal, for example, to bring forward those some stations, and also in Eastern Europe we are missing some stations. But still, you have a decent coverage over some parts of Europe, and all of those data are freely available to you as well. So go to the ICOS Carbon Data portal, and then you can download the time series for those stations. Okay, so I'm hurrying up a little bit. For us, it was a long-term dream that we would have continuous measurements of radiocarbon in the atmosphere. And until now, that was not feasible. Yesterday, we heard from Zavero that now that might change in the near future. Until that is the case, we try to find a way to work around that. And Ingeborg started with that shortly before the 2000s, when she introduced carbon monoxide as a proxy for fossil fuel CO2. The idea behind it is that if you have the combustion of a fossil fuel source, you also produce at the same time carbon monoxide. Yeah? And the advantage is that carbon monoxide can be very easily measured at a high temporal resolution and continuously. So now if you find a correlation between fossil fuel enhancement and CO2 and CO enhancement, you can use that ratio to estimate fossil fuel concentrations. And that is what we are still doing now. And here I show you a couple of the results from the last year. What you see on the x-axis is a radiocarbon-based fossil fuel enhancement measured in Heidelberg in flasks. And on the y-axis, you see the CO enhancement for the same flask. And we get a decent correlation for that. That was basically the idea which Ingeborg already had in the beginning of the 2000s. And we continued that work also for other proxies. So here you see the correlation with NOx. And we also looked into APO, which is atmospheric potential oxygen at the same time. So oxygen of being a tracer for fossil fuel, since if you burn the fossil fuel, you consume oxygen from the atmosphere. So if you then do very precise measurements, and if you factor out all the biogenic changes of oxygen, that's why it's called atmospheric potential oxygen, you are left with the change of the oxygen signal due to the fossil fuel or the ocean fluxes. But in Heidelberg, there is no ocean. So in Heidelberg, the cause for this change is the fossil fuel combustion. If you want to know more about that, we have to talk afterwards. With those correlations and the continuous measurements of those traces, we are now able to produce different fossil fuel estimates for Heidelberg. So CO-based, NOx-based, APO-based, and you see they are similar, but they also have distinct differences. And the idea is, what we are now doing, is we are delivering these three fossil fuel estimates to our Bayesian inversion algorithm in order to determine the fossil fuel fluxes around Heidelberg to a higher, with a higher degree of certainty. So these were just a few spotlights of Ingeborg's work, and we now see the full picture. So there were much more European projects where she worked on harmonizing the greenhouse gas observations within the European community, which then ultimately led to the establishment of ICOS, because it was clear that we need a coordinated and a harmonized framework to go on with the observations in the future. So, and with that, I'm coming to the end, and I just summarize what Ingeborg was for me and maybe for many others. And I think she was a pioneer in global radiocarbon science. For sure, she was not the first who started atmospheric radiocarbon observations. Yeah, there were others who did that before, but she was there when most of the others stopped the observations of the atmosphere. So in the late 80s, in the 90s, it was only the Heidelberg network which was producing global data, apart from New Zealand and other points, but network-wise, it was the only network. I learned after she passed away that she was a great role model for female scientists, so she inspired a lot of women in academia to really become a scientist as well. She was the mother of ICOS in all aspects, so she really pushed it forward, and it was always basically her baby, and she cared about it over the time when ICOS was actually growing up. She was fighting quite a lot for the integration of the different communities. And in the greenhouse gas community, we have the people who do the observations, and we have the modelers. And the modelers usually take the observations, do great science with it, and that's it. And she was fighting very much for an integration between these two communities, and she always forced modelers, you have to give your results back to the observational people, because that's what they need in order to stay motivated to do those good observations for you. And I would say we have come quite a long way in that, and the two communities are very well cooperating nowadays. She was a demanding teacher, that's for sure, but she left really a couple of good scientists who are still today in academia and continuing the work which she had started. And last but not least, she was a valued and a very honest friend to many of us who had the chance to know her in more detail. So for me, it just remains to say thank you to Ingeborg and for all of the things which she did for the community. And if you want to have a more personal view, we did set up a CUDA board after she passed away. And there you see what many scientists of the world did write about her and her accomplishments after she passed away. So if you have time and if you're interested, just scroll through that page, and you can also add your own words if you want. And with that, I'm done. Thank you.
Okay, thank you. Thank you, Samuel, for bringing her again to us. She will not be forgotten. We promise. The community always keep the big people in the heart and in mind. As I said, there is not a time for question and answer, but a minute of reflection. We can stand and have a minute of silence. Thank you.
So, Ingeborg always loved singing, and she was in a choir, so I think it's now to all of us to start. Volare, oh, cantare, oh.
Okay, so now I will ask the second presenter, Irka Haidas, to present what he wrote or what she wrote in the, or found in the Liv's memoir.
Okay, yes, I found the button. Good morning, everybody, and it is always difficult to speak after such a presentation. And I relate to the presentation because I was lucky to know Ingeborg personally for years. She would come to Hengeberg, and I think it's a fitting picture of Hengeberg trees. She was very supportive also with sharing data. So really, I add my personal thanks to this. Starting with the title page, you see all the co-authors on this presentation. Our technicians, Andre and Celine, actually a student working in the lab, and Urs is here, and the laboratory of ion beam physics, all the past and present technicians, engineers, and heads of the lab as well. This is a support from the lab, very substantial, because there is no funds for studies of students. Students who did the work, and school pupils as well, and the tree. This tree is still there, the very reddish one. It's a very special tree, Persian iron wood, that is actually not very common in Switzerland. So I will move forward and bring this slide just to make an introduction and maybe a content of my presentation and a motivation for this study. Many of you know the paper by Heather Graven that highlighted the problem of Suez effect overprinting the atmospheric bomb peak in our records. And that was 2015. And it made a shockwave among archaeologists, art researchers. Not that they studied that paper, but there was a Spiegel Online article that I have translated, not me, Google Translate, in English. It says, yeah, the radiocarbon relies very much on, archaeologists rely very much on radiocarbon method, but look at this, it's wrong. As far as I understand German, it is not saying in the future it will be wrong for archaeologists. So this online publication was, let's say, kind of a motivation for me to look at, observe how it is happening, and of course collect data for people to explain what's going on. Why? Why is this important? And Walter is here, maybe. He showed us many times how important the bomb peak is with his paper presented in Gliwice conference. He summarizes all these applications that are so relying on the bomb peak. I made a small figure with Google search for the publications of bomb peak. You can see how it grows with time, starting from the first observations, and if we make modeling for the next decade or completing this decade, maybe it will be flattening out, but hopefully this conference will have many papers as well. So let's hope we are on a rising line. Why is this important? It's just a headline to say for geochronology, of course it is important, but I will go for the pointer here, sorry. Oh. How do I make my pointer? No. With the mouse. Okay, you can see with the mouse here. For geochronology, it's maybe not such a big problem, because if the cores are continuous, we can kind of observe the bomb peak in the middle, sandwich with the older sediments. This is sometime in the future. But there are other applications, and just to acknowledge collaboration with this group here, with Gianluca, their paper on forensics application of bomb pig for ivory tracking of illegal trade. You can see this is disappearing, the bomb pig that is gone. It's a sad reality for forensic applications. Even other animals that we want to protect, you might see that there is an application of the bomb pig pointed out by Mebius-Guy already 2001. He had this very interesting paper on application of bomb pig to tracking illegal trade of wild animals. And this is just to point out, this is from the Swiss governmental webpage, where I know of the problem that the Swiss customs have. They have quite big collections of this chateau shell. If anyone comes to you to track down the bomb pig on this is from those Tibetan antelopes that are suffering, or the population is declining. So you see here, bomb pig is very valuable. Of course, for paintings, I can talk about this for hours. So if anyone interested in making Maleviches, talk to me. You can see here how bomb pig was and still is, for paintings like this, very, very interesting tool. So with the paper by Heather, the issue came up. We know that some and other scientists working on collecting data, modeling, they knew for years that it will happen, the bomb pig will disappear. But with the paper of Heather, we got this message through. And I started collecting leaves for looking at us. Maybe one can call this as a hobby, but I do teaching. I teach students, school puppies as well. We have small projects with them. I think it's an easy hands-on exercise, and I can use the data to show it to art researchers or archaeologists, explain to them this. And of course, the memories is something we cherish, and we should cherish more and more. So this is it looks okay on the screen here, but it's not a full map of all the locations where I have leaves collected, sometimes by me, sometimes by colleagues. I would have a huge list of people who sent me leaves. You can see here I have leaves, or maybe not yet on this map, from Australia and USA also. But yeah, the same kind of problem, big concentration of leaves in this part, although here is Vilnius and some leaves in Poland, so we're going eastward as well. And Africa, this is actually the point here in Ghana, is the very first leaf that a student from molecular biology brought to me. She was in Ghana for Christmas, and she brought me very first leaf. Very simple preparation, just an acid to remove carbonates, because there is no point in doing the whole ABBA, and simple preparation allows students to do it quickly, so you can complete the preparation in one day, including a collection of leaves as well. So the project can be completed in a couple days. And these are the results. You recognize the tree, co-author tree, Persian ironwood, Parotia persica, as they call it. As you can see it around in pictures, it's a different time of the year. I would collect actually It's not very systematic research, but I would collect a leaf in a spring and in fall. Although the tree is in here, I cannot tell you which of them, but probably most of those which are the closest to the green data of Jungfrau Joch, green carve. You see the wide spread of the points. This is all at Hengeberg, all at our campus. Students went around, they collected leaves behind different strange buildings. So you can see how diverse the C14 concentration can be in this actually very clean environment of ETH Hengeberg. This is a bit cleaner environment. This is the countryside. I'm lucky to live in this very clean environment of, you can see the place, Boppelsen, nobody knows where it is, even in Switzerland. But you can see on the C14 measurements that it is very clean to Jungfrau Joch. And actually some points, this point for example is from the middle of the village where the road is going by. It's a rose that I collected. So yeah, even the leaves can tell us how the atmosphere is changing. For comparison, Switzerland, still Boppelsen and ETH is there, but I put CH for Switzerland to the retrospective to see what's happening. This is Zurich. One of the students lives close to the very busy roads, and this is one of the leaves that he collected not long time ago. This is highway, not far away from Zurich, and this point, actually I have maybe name, no, later. I will explain this point because that's why the scale is so long to show this point. Compared to other countries, you can see here this very, very low one. It's Bangkok. Montana, our previous technician, she was going home for holidays to Bangkok. And you can see here, this is all collected in April 2017. She was in Pakchong. This is some kind of natural, I never went there, but on the pictures you can see how green the place is. So few of those points sitting on the green Jungfrau Joch carve are from Pakchong, but Bangkok, see how old it is. The leaf, I cannot tell you exactly from which part of Bangkok it is. I have to contact Montana again. And now I have explanation for this point. It's a wedding flower from one of the technicians. I believe she remembers her year of the wedding, but see that it doesn't really fit. So I suspect this must be some greenhouse effect of growing trees or flowers flown from, I don't know, Netherlands. So you see, I'm sorry, I have fun looking at these points. It's not very... So that is pointing to, I think I have more pictures. All right, this is Trieste. Here, this one. That's Trieste station in the back here last year. You can see how low it is. So more pictures, Vilnius there and some other towns. And yes, we're talking about memories, but this is kind of a trailer. You can see we will go to this, maybe they will squeeze us into this room. And it's not very sharp because Gianluca is moving. He is never changing, you know. If we take the picture in 10 years, he will look like this again. But that is what we are, I think, pretty soon we will measure sample. This leaf collected at the tree, see in front of the big wall as you walk around, the tree of magnolia, we think. This sample we have, and then there is this plant, and there is a competition now. Do you know what age we will get from this plant? So I think that was the last of my scientific slides, and this is my closing remarks. I cannot call it conclusions, really, because, these are just ideas. I think measuring leaves, we can get the information already without a big equipment. You can just collect leaves and use it for teaching purposes. If we don't have funds, we can really educate new scientists, new generations, or even, and I put this potential citizen science, I don't know why with the question mark, because I'm also not so clear how one could push this forward, but I think people looking at the contamination around them, they would be more aware of the greenhouse that is coming, not this old. This one is definitely more than 40,000, but I have another birch leaf from Krapina, which is across from here. So thank you very much. And
thank you, Irka. You want to sing? You want to sing? No. Volare. Oh, what? Okay.
No.
Thank you for the wonderful presentation. Wait. I have a question by myself. Should we send you a letter with a leaf when we go back home?
Well, that might be. I think many conferences I
So it means yes.
There may be a huge pile of samples. I don't know if by the next, depends how many students I have. There is a next group coming.
Okay, so now we have a time for a few questions, and Alex was the first.
Another example which was done in the middle of 1970s. I got from green leaves in downtown Moscow. It was 500-year-old leaves. And 150 kilometers north, 134% of mother. That's a huge difference. It's about 40 PMC difference at the same time. And I don't know what will do future archaeologists where they take samples.
That is exactly that problem that archaeologists kind of don't realize.
Yes, Kosiara?
Oh,
I still have 1 minute on my time. Yeah.
is that okay?
Yes.
It's a great idea, Jirka. My question is that from the collection that you were showing, the results that you were showing, was those from perennial or annual?
Annual.
Okay,
Yeah, but
you.
actually, I have a quite data set of needles from clean environments, and this would be kind of a suggestion for anybody working with bomb peak reconstruction that there are clean archives of clean leaves somewhere. We just have to go out and contact these people that they have those. And in this way, we would be able to maybe fill them up better with real data. I mean, this is just idea data, what I'm saying, but yeah. No, these are annual. But the students, I didn't go to details, obviously. The students went so that they sampled, for example, around the parking exhaust place, and you can see it. you have to measure precise, I mean reasonably precise, but you can see the difference. You can see how the C14 signal is diminishing closer to the parking lot.
To the source. No,
Yeah.
I believe
Yeah.
you. It's just that when Andre is asking for us to send samples to
Okay,
be
yeah.
careful that
Yeah.
they have to be from
And possibly
annual
on the height of,
vegetation.
not on the ground
Yeah.
as we saw.
So
from
Okay.
maybe
other-
send a little protocol, or what type of samples should be, and something like that.
And send me the location in email, please.
Okay, thank you. So, I mean, no more question. Ado, you have an answer or punctual coffee? So, no both in the same time. Okay? So we have to pass to another presenter. It will be Mishi, and he will give us the up-to-stage information about the Heckel laboratory, or not.
Yes. Okay, thank you very much. Good morning, everybody. It's a great honor to be here in this session after these great talks. Thank you very much for the opportunity. Today I'm talking about our first attempts to try the atmospheric methane radiocarbon measurements. We did this work in a close collaboration with the IMAU laboratory from Utrecht, the Netherlands, and many of my colleagues. Maybe at the end of the presentation you will understand why the group is so big. So first of all, we already learned also today from Sam Hammer's talk that methane is so important. Methane is the second has a second importance after radiocarbon in the greenhouse gas effect. It is much less in the air, but much stronger greenhouse gas than CO2. So, and methane level is rising constantly. Actually, the rise in methane concentration was more dramatic than in CO2. It more than doubled, close to 3 times more than before the industrial area. And also, methane concentration is doing funny things. For example, around 2000, it stopped rising for a few years, and then it rises again. So it's not that straightforward to understand, okay, we are releasing methane and it is rising. It is not that easy. As we learned on Tuesday from Quam Hua, that the source distribution of methane is very complex. It has natural and anthropogenic components, and anthropogenic components can be bio and fossil. So it is a little bit tricky. That's why that scientific communities has to use isotopes to understand the whole picture. The biogenic, the pyrogenic, and thermogenic methane, all of three has anthropogenic and natural origin. And unfortunately, stable isotope, at least carbon stable isotope alone, doesn't give the resolution of the picture. It is not enough diverse. They are too uniform to be distinguished. If you combine it with a hydrogen stable isotope, the picture is still too complex to understand. So they are now using lambda isotopes also from methane and others. So it is natural wish and to use also radiocarbon to understand at least the proportion between the fossil and biogenic portion, as it was initiated by Ingeborg Levin, as we heard this morning. As it works for CO2, it should also work for methane. So it would be very good to measure the methane, atmospheric radiocarbon in the atmospheric methane. Actually, it was started, not the atmospheric, but observation of the difference between bio and fossil methane was done by Libby, by gas proportion accounting. And by the late 70s and 80s, there were many studies showing how to use radiocarbon to understand the methane sources. It's a pity, or it is complicating even the picture that nuclear power plants are emitting radiocarbon, methane to the atmosphere, which is elevating the background. It is not the bomb carbon, it is a continuous release from nuclear power plants. So it rises a lot. It has an oxidation process in the atmosphere along with a 10 years lifetime. So the whole picture is quite complicated. And if you look into the literature, you can see that very little number of atmospheric methane radiocarbon data are reported due to many reasons. First of all, you need a really high volume sample, a few cubic meter of air if you want to have a 1 milligram sample. So it's not that easy to collect. For a certain research project, yes, but for continuous monitoring, for example, was done for CO2, it is pretty impossible at the moment, I would say. And there are other complications, and carbon monoxide is less, but not that much less than methane, about 10%, and it behaves very similar in the extraction lines. So you need something to remove CO2. There are tricks to do this, but at least it is one more complication. And unfortunately, methane cannot be freezed out or simply in a glass or steel trap with liquid nitrogen, which is also another headache, let's say. And at the end, most AMS system needs at least CO2 or graphite, so the methane has to be converted. So there are many problems with that. So it was enough for us to say, okay, let's try this. Let's try the methane atmospheric measurements at our laboratory. And we tried it do it as simple as possible, because the target will be to mass production of atmospheric methane data. So you would need something simple. So we tried a very simple approach in the garden of our institute, which is a city background site for atmospheric aerosol and CO2 radiocarbon measurements for decades. So we tried a simple filter, drying, a membrane pump, and a not very cheap, but available bag for gas samples. It is less than 30 liters. It was a bigger sampling campaign for stable isotopes, but we used the same gas for testing the concept. So the concentration was measured by the Picaro cavity ring down. The stable isotope mostly measured at IMAU research lab in the Netherlands. And we measured some radiocarbon data at home in Debrecen. There were many complications, for example, to move a few cubic meter air from Debrecen to Utrecht. That was a lot of fun. So following the methane extraction and purification for stable isotopes at Utrecht, we were able to use the extraction and purification line to have a very strong quality control to have really clean methane, not CO2, not CO methane. This was the first, I would say this is the hardest part. And we were relying on the knowledge and expertise at IMAU in the Netherlands. And then the other problem to move the tiny methane samples, which is the range of 10, 20, 30 microgram carbon from that lab to our lab, and do the other stuff, the combustion and measurement of methane. So the first part at IMAU, we were using their line, but it was for a few cubic meter of air, not tens of liters of air. So we eliminated the bigger traps where they trap the water and the CO2 from a huge amount of air. We use just a much smaller, on the bottom, simple spiral glass tube. It was enough for 20 liters of water, air. But the other part we need, so we use the huge first charcoal trap and also the second one for focusing the methane. It is not completely pure at that stage. It has nitrogen in it at the end and other, maybe noble gases, other stuff, but it is very much concentrated. It is then a few cubic centimeter maximum. And then this amount of gas is going through a GC line, which is behind Balázs, my student. It is completely clean. And at the end, we can collect the methane. Collecting methane, as I thought, is not that straightforward. So we are collecting methane on small grains of silica gel. It works like a zeolite. It's trapping the methane on the liquid nitrogen temperature. So we put a few or maybe one silica gel on the bottom of a regular reservoir tube, 6 millimeter, and freezing the methane there. And then you can seal it and move to the Debrecen lab. So then in our vacuum line, we transfer from one tube, the reservoir tube, to the other, the bigger 10 millimeter diameter tube with manganese oxide. In all applications in our lab, we combust with manganese oxide. It's much easier than copper oxide. You don't need quartz. It's very clean. So we put also a grain of, you need to freeze that methane, and it is not freezing on manganese oxide. You need the silica gel. So we freeze it over, freeze the methane and close, and it is a sealed tube combustion. So theoretically, you can combust 100 samples in a night if you want. So we put it into the oven. We have the CO2, and we have this smart line when upside down with this little bump at the bottom of the tube. Oops. There. So now it is upside down with a O-ring sealing. We open the tube, freeze out the water, and then it is CO2, the business what we do every day. So then it is, let's say, easy. And the CO2 is going to be graphitized if it is more than 100 microgram. The graphitization tube, the last but one, with again a seal tube graphitization, what we are using for almost all the applications. Or if it is too little to be graphitized or to be measured in a graphite form, then we move it 4 diameter, 6 centimeter long reservoir tube, which you can see on the right side. And these are the ready samples for the gas ion source measurement. Oops, sorry. And then we use our Lecce system for the gas measurement because it works very well. The Lecce for really microsample measurements, it works super well. So we break the tubes in the cracker system here and flush it with helium to the syringe to have the 5% CO2 in helium mixture, and the syringe is open according to size. The maximum capacity is 140 microgram, but you can even measure 5 microgram. This is an example of a blank measurement on a 5 microgram CO2 blank sample. It is meaningful measurement. So we have this tool to measure the tiny methane origin. At the end it is CO2, but it was methane sample. Okay, so then because the expected sample, sample amount is maybe 10, 20 microgram, we had to test all the steps. First of all, we tried how well we can measure a few microgram of carbon with the gas source, and it works very well. So maybe the blank level is not 1 PMC but 2, but the OXA is giving the same signal, just a much bigger error because of the short measurement. Then we tested the combustion blank, which gives after blank correction about 1 PMC answer, so it is really much less than 1 microgram modern carbon contamination. And then we also tried to move fossil, a split of fossil methane from the IMAU line, but not processed, with all the steps to the lab, and it was about 2 PMC, so it is still not very serious background on the size of 20 microgram sample. And the most important thing, when you start from the very beginning, from the extraction line, reconcentration, GC, and everything, and come to Debrecen and measure the radiocarbon, and then on a 8 microgram sample, we have seen 0.25 fraction modern, and close to 20, it is about 10, 15. So it corresponds to 2 microgram, 2.5 microgram contamination in the whole process. It is not perfect yet, but it is not very far what we want. So we need to improve. Maybe we made only little modifications in the prep line. We need more modifications to optimize such a small volume, which is 100 times less than their usual. And then we were ready to do the first test measurements in Debrecen on atmospheric methane. The first few samples are the very easy ones because they were concentrated. The concentrations here are higher. The size of the methane sample was between 100 and 200 microgram, so it is easy for the GIS measurement and for the whole process. And we have measured the coal farm, which is very, very close to 1 modern. The wastewater biogas outlets are a little bit below. Landfill covered landfill is above 100 PMC, 1 modern, because it has bomb peak original organic carbon in it. And open, more fresh landfills are giving a slightly below 1 modern radiocarbon signal. Just for testing, we run also the natural gas in Hungary, what we measured, not only the standards from IMAU, and also thermal water methane degassing, which was very close to fossil. So it was also a verification that the work is clean, what we are doing there. So now finally, at the end, I can show just a few atmospheric methane radiocarbon data from Debrecen from different times, one from the spring, more from the summer, and in the autumn. It was 20-liter sample, and the samples are scattering a little bit between 1.2 and 1.4 fraction modern, with quite big errors at the moment. Is due to the large blank correction, and maybe we are too optimistic to use only 20 liters of gas. Maybe we need 40 or 60. We will see in the future, but at least we have the first few data, and it is very promising. So we think it's worth to continue. We will optimize the line further to investigate the constant contamination. We have to stabilize the carbon recovery because we have seen some anomalies in the mass balance, and a few repeatability tests, and maybe we use a little bit bigger bags But in principle, it will be possible to do atmospheric methane measurements also in our laboratory. So thank you for your attention. Here you can see the group with IMAU and us. We made a big goulash soup lunch at IMAU, and I believe it really helped the methane studies.
Thank you. Do you want the boys band to come to sing?
We did it yesterday.
Okay,
Everybody
so once is
remembers
enough.
the
So
Backstreet
we have a time for one short question, and I will be the one who choose the person who. No question. Thank you. So okay, Walter. Yeah.
At the beginning, you showed a paper which compares methane contribution from agricultural and fossil fuel.
Yes.
Is there any trend visible? Which of the contributions will be larger in the future?
There must be, but I don't know. I cannot tell at the moment. There are really serious big studies about that, and they are really waiting for more radiocarbon data. So they need more and more help to resolve this complex source and sinks structure.
Okay.
And concerning the ratio of the concentrations of CO2 and methane, right now, if you include the global warming factor for methane, which is a factor of 20 larger per molecule than CO2, and take the concentration into consideration, then you actually get still a factor of 10 between CO2 and methane for the contribution to the
Yes.
global warming effect. But this may change actually as time goes on.
Yeah. That's why would be very important.
Okay.
Okay, thank you. So now I would like to invite the next speaker, Christina Eriksson. She will show us what's going on in the Baltikum around the nuclear power plant.
So, good morning. This morning we've heard that that the carbon-14 levels in the terrestrial environment can be affected by the fossil fuel emissions, and we also heard in the last talk that we have nuclear power plants raising the carbon-14 levels. And I will now talk a bit more about the nuclear power plants, and I will give you an example from Sweden, and I will not be in the terrestrial environment, but I will move down into the marine environment instead. So, nuclear power plants are known to emit carbon-14, both to air, but also to water, during normal operation. And it is important to track these emissions, and this comes because carbon-14 is known to often dominate the committed effective dose to the public due to these radioactive releases. And the legislation is very strict in this regard. In Sweden, the legislation says that no member of the public should receive more than 0.1 millisievert per year from radioactive effluents from nuclear power plants, and that includes, of course, this carbon-14. For releases to air in Sweden, carbon-14 is measured both as methane and as oxidized carbon-14 routinely, and that has been done since 2002. And if you look in the terrestrial environment of Swedish nuclear power plant, you can find an excess in biota of about a few percent, up to perhaps maximum 10%. And this is fairly well known since measurements are actually being done. But when it comes to discharges to water, it is known that these are significantly lower than the airborne releases, but in most countries, these releases or discharges to water are not measured because they are considered to be low. But it must be remembered that the spread of C14 into the atmosphere is quite different from what happens in the ocean. You don't get the same dispersion at all in waters as you do in atmosphere. We have done a few measurements in the past around some nuclear power plants in Sweden, but not much. So the environmental fate of carbon-14 discharge to water is fairly unknown, I would say. Our research group at Lund University in Sweden, we have used seaweeds as bioindicators in many studies concerning other radionuclides than carbon-14 for quite some years now. And lately, we have been making extra efforts to also include carbon-14 into these studies. To give you a bit of a background to what I will show you now, we did a study a few years ago and published in 2022. And in that study, we looked at spatial and temporal variations of carbon-14 in seaweeds, fucus, around the Swedish coast. And this study was, and it still is, financed by the Swedish Radiation Safety Authority. And what we wanted to find out was how are the natural variations of carbon-14 in the marine environment, and what kind of anthropogenic influences do we have? Here I show you some results from that study. And we started collecting samples. Let's see if I can point here, perhaps with this one. Perhaps you see up here, there, we have sample site 1 that is close to the border of Norway. Then we moved south into the Baltic Sea, and then moved up into the Gulf of Bothnia, and we collected seaweeds. That was fun, I could say. I did the sampling. And in this graph showing you the results, this dashed line, that's the average of 26 terrestrial samples collected in Sweden, in southern Sweden in 2020. And I would like to point out a few interesting features in this graph, and I'm sure you see them. First of all, there is a peak, quite some peak actually, and that is around one nuclear power plant on the Swedish west coast, Ringhals. And of course, this is interesting. Another interesting feature that you can see is that there seems to be a gradient in the carbon-14 values decreasing from north to south. Of course, one wonders, what is this? How much of this slope that you see here, this gradient, is due to Ringhals nuclear power plant, and how much could be due to other sources? And, of course, these rather high levels that we saw at Ringhals nuclear power plant in seaweed, what is the local radioecology there? How about other species living in this environment? First, when it comes to this gradient that we saw from north to south, we are pretty convinced that the main reason for that is these reprocessing plants for spent nuclear fuels that are located here in France, Le Hague, and Sellafield in the UK. And C14 is transported with ocean currents up here to the Swedish west coast. I just want to show you a very quick picture of this, and this is another series that we've done. We have one professor in our research group who has collected seaweeds since 1967, actually, and he's done this every second month or so. So we have a large biobank of fucus, or seaweed samples, and we have measured carbon-14 in these samples. And in the lower graph here, there is the pointer, these brown dots are the C14 levels. And you can see that here in the late 1990s, there is suddenly a rise. In this upper graph, I've plotted the reported discharges of carbon-14 to the ocean from Sellafield and from Le Hague. Those are publicly available. And then I've applied some delay time and also some dilution factors that we found from other studies using technetium 99. And then you can see this blue graph up here, it's a sum of the two. And you see it coincides very well here in the late 1990s. We have this big peak, and that is we are pretty sure what we see here as well. So what about Ringhals nuclear power plant and this local radioecology? First of all, Ringhals nuclear power plant has had 4 nuclear reactors in operation. 3 of them are being pressurized water reactors, PVRs, and 1 is a boiling water reactor. Today, only 2 of these are still in operation. 1 was closed down in 2019. One of the PVRs and one of the BVRs was closed down at the end of 2020. But we still have 2 operating nuclear power plants, reactors here. We heard in the previous talk that when it comes to pressurized water reactors, and I can also say that these are the type of reactors that are most frequent in the world, they release carbon-14 mainly as hydrocarbons, some carbon-14 but mainly hydrocarbons. And you see the release rates here. They are rather high, like per reactor it's about 200 gigabecquerels released into air. Boiling water reactors, they mainly release carbon-14 as carbon dioxide, and often with about twice the release rate as you have from a pressurized water reactor. And just to show you these data that the nuclear power plants have reported, you can see in the graph down here, you see how much is released in oxidized form as carbon dioxide and how much is released in reduced form, mainly methane. And you can see here that in total from Ringhals nuclear power plant, there was when these measurements started, about 1 terabecquerel per year. And then you can also see here that towards when the BVR shuts down here 2020, then these green bars go down, and that's because the BVR has shut down, not releasing so much carbon dioxide anymore. So what about the releases to water then? Well, it's fairly unknown, as I said, but it's believed that from pressurized water reactors, it's mainly as inorganic carbon-14 that the discharges are. But there could be some organics too. And those chemicals forms are not well known. It's been speculated if it's alcohols, aldehydes, different hydrocarbons, but it's not, as far as I know, there's been no investigations of the chemical forms of the releases. And the amount released to the environment, well, there are not much reported. We did some studies in the past which pointed to that it would probably be less than 1% of the atmospheric releases. But I recently saw that nuclear power plants in France actually do report their discharges. And if you look at those data, you come up to about 10% of the atmospheric discharges that goes into water. When it comes to boiling water reactors, this is believed to be mainly as organic, and it's believed to be less C14 to the marine environment from BVRs than from PVRs. So, what have we been doing in this radioecological project? Well, it's ongoing, and the results I will show you now, it's as far as we've come at this moment. We've done an intensified 1-year study at Ringhals, and we've looked at seaweeds. And seaweeds, they are good at absorbing dissolved inorganic carbon from the water. But we have also looked at fish, and a fish called cork-wing wrasse. We've looked at mussels. We looked at soft tissues. We looked at shells. And in the next talk, you will hear more about the shells. We've looked at oysters, and then we didn't look at the shells, but at the soft tissue. We looked at crabs, and also at biofouling plates. And these I will show you some results of. We have also collected samples of particulate organic carbon in seawater and sediment, but those results are not yet ready yet. So the whole idea is that we want to fit what we measure into some kind of radioecological model. And this is one model, how it could look when it comes to marine carbon-14 discharges of dissolved inorganic carbon. The organic carbon is not in here yet, but this is for the dissolved inorganic carbons. And I've tried to fit in here where the different samples that we collect in what kind of, where we find them. And perhaps a mussel here should instead, of course, it's a filtering organism, but it's, of course, here in the marine food web. So, some results that we have. Just to compare, we also collected some grass samples close to the nuclear power plants and also at other sites along the Swedish coast. But and we also have one reference site in the Baltic Sea. This is Skillinge, it's called, so it's not on the west coast anymore. And if you look at the average here, well, perhaps I should first say that Ringhals nuclear power plant is here, and then the water in which the discharges are, it goes through tunnels that are dug here into the ground, and then this red arrow represents where the discharges are into the sea, and it's in tunnels. They are about 5 meters below the surface of the water. Anyway, we don't see much in the grass at all. Actually, it's not significantly higher than at the reference sites. And of course, the two operating reactors are pressurized water reactors, and then it's mainly methane that is released through air, and that is not taken up by the plant. So it's no big surprise that we don't see anything now when we only have pressurized water reactors. But what about these biofouling plates then? Well, here are quite some high results. These biofouling plates, it's plates of plexiglass that are located just at very close to the outlet of the discharge outlet of the cooling water. And all kind of organisms grow on these plates, both animals and also different kind of plants. And as you see here, at one of these biofouling plates had a fraction modern of over 8, and that's quite high, I would say, surprisingly high. But how about the living organisms then? Well, here you can see some results from spring last year and the results from brown algae. And as you can see here, the highest levels, where is that? You see them here, close to the outlet. It's not so big surprise, really. Fraction modern of 1.8 or something. And then it appears that the cooling water plume moves south first. So we have some sites here where we have elevated values. And then from other studies that we've seen, the plume of water moves north because there are strong northerly currents in this area. The next type of organism we looked at are filter feeders, and then we looked at both oysters, these Pacific oysters, an invasive species, very fast growing, and blue mussels. And what is interesting here, I think, is that we see that close to the outlet, the oyster collected there, it's this thing here, it has very similar values to seaweed, but the mussels, these blue mussels collected down here, they are actually enriched in C14 compared to the seaweed. And that also goes for the other station down here. For crabs, we see some elevation. They don't vary much. But for fish, it looks quite different. We can see that it varies very much between sites, and obviously these fish are swimming around and have a large habitat because we found the highest levels down here, up to almost 2.5 C14. And just to show you some results from autumn 2023, we find even higher values of carbon-14, especially in fish. We have more fish samples not analyzed yet, but you see here, this single fish had fraction modern of 3.5, and that's high. But some interesting features, I think, and I would be happy if you have some ideas why it looks like this, because I'm a bit puzzled. I have some ideas, but We see that we have similar values in seaweed and oysters. Why is that? We see that mussels obviously have higher concentrations than both seaweed and oysters. I could buy that because the filtrators, it could be the organic part of the ecosystem. And then this, why are the fish so high? That could be really interesting to find out. Some ideas, and I will just conclude very briefly here what we've seen. So we've seen very large spatial variations in the area and high values, I would say, surprisingly high. I didn't expect this when I started the study, that we would find up to fraction more than 8 in these biofouling plates. We don't see much variation in crabs, for instance. They are rather stationary, they don't move around much, so that is not surprising either. But we see a large variation in the fish swimming around, perhaps not surprising. And we see these differences between species that I find is the most challenging to explain. I have some ideas, perhaps, and I would love to hear your views on this. Of course, it could be that we have had higher discharges in the past that are now stored in sediments and also in the marine ecosystem at various parts. It could also perhaps have to do something with the organic discharges, that they are actually perhaps there are like bacteria who could oxidize these organic discharges and in that way come into the ecosystem, being absorbed by mussels, being eaten by fish and so on. So perhaps we need to also include this in this part of the whole ecosystem. What happens with these dissolved organic discharges of C14. But I would love if you could approach me after this with discussions. And I would like to finish with a song of the cork-wing wrasse, the fish. And it's in Italian, so we'll see if you understand what I mean. So I say, nuotare, oh, mangiare, oh. Thank you. So for those not speaking Italian, it was swimming and eating.
Yes, this is what we love.
Yeah.
Yes, and thanks for this announcement. So we don't have a time for a question, but the next presentation is related to this,
yes.
And maybe Christina can join if there will be a question.
Yep. Thank you.
So we have to stick to time. Tick tock, tick tock.
The other microphone.
Yes.
So I can walk
Yes.
around.
Please, you can walk.
Thank you.
Yeah.
No.
So this will be about the blue mussel and if it is safe to eat them, maybe, or not.
I'm going to talk about the shells. I don't think you want to eat the mussel shells.
Oh, okay. But the inside?
You'll see. So my name is Sofia Bjarheim, and I'm going to talk about the potential of blue mussel shells for retrospective analysis of marine carbon-14 discharge from nuclear power plants. Quite a mouthful, isn't it? This is what I did for my bachelor project at Lund University, where Kristina Stenström was my supervisor. And I'm currently just started a master's in physical oceanography in Gothenburg, Sweden. So why are we doing this? Kristina talked about the emissions of carbon-14 into water and that the nuclear power plant should start measuring this. But there's no good data in the past. We wanted to be able to recover this, and therefore we chose to use the blue mussels. They, at least from the sources we found, are made of dissolved inorganic carbon, the shells. And the shells also have annual patterns, meaning that if you pick a live blue mussel shell and divide it according to the pattern, you should be able to send in a sample for a specific year back in time. Past similar studies, a lot of people have used Arctica islandica and other mussel to look at the bomb pulse. And there's also studies where blue mussels have been used to look at carbon-14, but then they're either looked at the soft tissue or the whole shell. We couldn't find any examples where they have divided the shell according to the annual structure. And of course, there's Christina's bigger project with the emissions along the coast, but they haven't looked at the blue mussel shells until now. The sample sites for the shells were Båteviken, up by the Norwegian border, Bua, by the nuclear power plant Ringhals, and the reference site in Særdal, all sites that Christina have previously used. So, before we go into the physics, you need to know some mussel words. You have and the blue mussels can be up to 20 years of age. So, the region in front, up here, is called umbo, and then you have the posterior edge beneath it. The longest line you can draw between this is the axis of maximal growth. What should also be noted is that the name for the different parts of the shell is depending on where the hinge of the shell is. For the blue mussel, the hinge is here. Therefore, if you have a more rounded shell, you often find the ventral edge opposite to the umbo, like we saw in the shells yesterday, if you were paid attention. And then you have different shell layers. We have the periostracum, which is a really thin organic layer on the outside. Then you have the fibrous layer, which is the blue part. The fibrous layer grows from the umbo and towards the posterior edge. The annual rings in the fibrous layer will be almost perpendicular to the curvature of the shell. Then we also have the nacre. It grows from the fibrous layer and in towards the mantle. The nacre will have annual rings that are more parallel to the curvature of the shell. And what I did for my project was to develop a method. We wanted to know, is this possible to do? The goal was to extract a sample from each annual band. And the first part of this was to locate the annual bands. We did this by first sanding off the periostracum, and then we put a bright light under the shells, and there you can see the annual bands as these darker rings in the shell. After we did this, we made something called an acetate peel. We saw the shell according to the axis of maximal growth, poured some acetate along the cut, and then pressed it against the cellulose film, and then you get an acetate peel, which looks like this. It's similar to a thin section, but it requires less work and it's easier to do. Lastly, we also compared the shell sizes to growth curves to estimate whether our assumed ages were reasonable or not. Then we needed to divide our shells. We had already cut out the triangular piece in the middle, like you see here, for the acetate peels. And then we marked the annual rings and cut the shells according to this, like the small pieces up here. As I mentioned, the annual bands or annual rings do not align in the nacre and the fibrous layer. Therefore, it was really important for us to only have samples from one of the layers, since we wanted the samples to be for only one specific year. To be able to separate the layers, we heated the shells. This, for the samples we sent in, we did after we had divided them into the small parts. But here I have a picture from the process or the method development, where we heated the entire middle section, and you can clearly see how the layers have separated. The samples from the fibrous layer were then sent into the carbon-14 lab in Lund for SSAMS analysis. And over to the results, the fun part. Here you have the same graph that Christina showed you, but now with our blue mussel results. And here is the zoomed-out version with the blue mussels and the seaweed. You can see that for one of our shells, the trend line is quite similar for shell number 1. This is really promising. It should also be noted that for this site, and at this time, there should be no other influences than from the bomb peak. And therefore, we assume that the carbon-14 in the seaweed, and the shells should be really similar at this time. However, you can see this is not quite true for the other shell. And here I want to point out that blue mussels are semi-sessile. It could have moved around or been washed away with the waves and ended up at this location. Because if you look at the last 2-3 years, it aligns quite well. Yeah. And what's this? Yeah. And next we have the results from Båtviken. This is the site by the Norwegian border. And these samples are from our day and age, it's not the old ones. In Båtviken, as Christina also talked about, we have the influence from La Hague and Sellafield. And they estimated that the emissions will appear in Særdal about 2 years later. We expect it to be a bit less for Båtviken. You can clearly see how the peaks from our blue mussel shells align quite well with the emissions from La Hague just 1 to 2 years later. This also shows the promise of our method. You can furthermore notice that the emissions or the carbon-14 in the water is higher than the grass samples down here, which again is due to emissions from La Hague and Sellafield. The grass itself, here you have both from Båtviken and the Bua site, follows the trend line for southern Sweden quite well. And yeah, there's also some seaweed samples for comparison in the plot. And now we have the most exciting part. It's the emissions and or the samples by Bua, the nuclear power plant. What's really interesting to see is the oldest shell sample with the high results of a fraction modern that's similar to the top of the bomb peak. As you can also see, the values jump up and down quite a bit year to year, so there's clearly something influencing the shells, right? However, our shell samples don't align super well. It could again be that one of the shells have moved around a little bit. We don't know. It's also quite hard to find the annual rings in the shell. So it could be that, for example, if we missed something here for the shell number 2, it could be that these 2 plots should be moved by year. It is possible that we mixed up our samples for one of the shells so it would align better. We don't think we did, but I mean, it could have happened, right? And yeah, you can also see how the samples from the seaweed have different values of carbon-14 year to year, so we do expect these temporal variations. And again, as Christina mentioned, 2 other reactors shut down 2020 and 2019, which again is clear. It's shown that the values decrease of the samples from 2020 and later on. Yeah. So moving on, our method is really promising. And for Særdal, the reference site where there should be no other influences, the blue mussel and the seaweed samples agree quite well. You can also see how the carbon-14 from the nuclear power plant in Ringhals at Bua does affect the marine environment. And in the future, if we continue or for future measurements, we want to be able to take more samples from each site, both to eliminate the possibility of the mussels being semi-sessile and having moved around, since while it's possible, it's not super common, and also to eliminate the uncertainty in estimating where the annual rings are, because bigger sample size, less overall error, right? And that's what I had to say, so thank you.
Thank you for the talk. You don't want to sing?
No, thank you.
Okay. Okay, so we have a time for a few questions. Juan? Yeah, Juan, please. You will be the next.
Okay, thanks for the talk. I have a naive question. Okay. Can you go back to the diagram you showed? Yeah. Okay. I assume, not assume, but the shell live a number of years, and you separate a single year for the shell.
Yes.
Right? But for the soft tissue, how do you know which year is? Because the thing is, say if the shell live about 5 or 10 years, and then when you collect the soft tissue, how you know this is 2022 or 2023?
Okay, yeah, so as you said, the shells, they can live up to 20 years. We have estimate, or we don't think they will get that old along the Swedish west coast due to predators. We talked to aquatic ecologist about this. He's also one of the co-collaborators for this. And with the soft tissue, we have assumed it to be from the year the shell was picked. These shells are actually picked in 2023, but in spring. And I have a backup slide for this. Here. So the shells, the annual ring for the blue mussels in Sweden is created in winter. And these shells were picked in middle of May, when they just have started growing. Here you can see a cutout of the acetate peel for one of the shells in Bua. And as you can see, there is an annual ring close to the edge with less than a millimeter to go. This we discovered, because when we did the shell division, we did it according to the external structure, as we found that the most easy way to go, and there was, and we weren't able to cut out this last millimeter from the year when the shell was picked. So therefore, we said that the mussel meat for the shells in Bua is from 2023, but the youngest shell sample is from 2022. For this pilot project, this does only apply to the shells from Bua, since the other shells were picked in autumn and winter.
Yes. So,
did that answer your question?
Yes, and Susanne?
Yeah, not to mention that I'm very fond of your talk and Christina's. Have you, because I know problems in identifying the years or the growth lines, have you tried using Mattweiss solution, which colors your shell blue and emphasizes a bit the lines because it edges away the soft parts? And second, what was my second? You mentioned you heated the shells to separate the layers. How high do you have to heat them? I've never done that, and it sounds absolutely interesting.
Okay, so I read about the Mattweiss solution, and well, sadly, since this was only a bachelor project, I had half a year and only like 50% studies for that half a year, and we didn't have the time. We also were a little bit scared about introducing too many chemicals without having the time to look into it, because we don't want to contaminate the shells. We also, from the study, or the little I read about Mattweiss solution, where they had used it, they looked more at the nacre, because for the blue mussels, since all of the annual rings in the nacre can be seen in the umbo, it's often easier to count the shell age there. And everything I read where they have determined the shell age of blue mussels, they haven't wanted to divide them and have mostly looked at the nacre, since that is easier. Mostly we didn't have time, but yeah. What was your the heating. So we heated the shells to 500 degrees Celsius for about 10 minutes, and that would separate the nacre and the fibrous layer. We weren't able to separate the different annual rings themselves, but apparently that has been achieved for some other shells. Bad thing is it becomes more brittle afterwards. And in my bachelor thesis, there is a longer discussion on the method. There's also a step-by-step process on how we did ourselves with the shell and the pre-treatment in the lab. So if you go to Lund University student paper and search for my name, you should be able to find the descriptive method.
Yeah, thank you. One more question? Yes, so Martin?
Yes, I wanted to ask maybe also to Christina about the discharge. Is there like a constant discharge of the power plant? So is there like an annual circulation or when the power plant shuts down for service? So that would, since you just showed like growth periods basically, could that affect the annual measurements then?
Well, again, this is hard to say since there's no measurement of carbon-14 in the water. And carbon-14 is a weak beta emitter. It's kind of hard to measure, especially when it's in low doses. We have these emissions into the air. And we try to look, because they have measured other isotopes, for example, tritium, into both water and air. For that, it didn't seem to correspond with the airborne releases and the water discharge, so it's hard to say if it relates. So, I mean, it could vary around the year and should, in a way, influence when we should pick the mussels and things like that, but we haven't had the time or possibility to investigate that further.
Okay, thank you. And we can now proceed to the last presentation about how to save or at least how to help to combat the traffic of ivory in Brazil using the radiocarbon method. And I still don't know what's your name. Caroline. What? Caroline? Yes? So Caroline Silva.
Yeah. So, the pointer is working? Yes. Okay, good morning. My name is Caroline Silva, and I am a PhD student, and I'm going to present to you my master's research. So the title is Radiocarbon Data and the Shoeda Partner as Essential Tools in Combating Elephant Ivory Trafficking in Brazil. So there is an overview of my work. In introduction, I'm going to talk about the goals of this project. And in the materials and methods, I will talking about the difficulties you have to do this work, about the protocol. I will present some results and the conclusion. So ivory is a highly estimated material that has been treated for centuries, initially usually for sculptures and later for bouquet curves or piano case. And the increasing illegal elephant push forces the establishment of an international law prohibiting the trade of a such material. So our laboratory, the Radiocarbon Laboratory of University of Federal Fluminense University, is the only one in the Brazil that contains the facilities to do the measurements since the treatment until the measurements in the accelerator. So Brazilian Federal Police contacts us to do a dating of this type of material, of the ivory samples, because right now there is a case of the traffic of this kind of material. So you have the sites, is the conventional or international trades in the regional special species of wild fauna and flora. And the site established a law in 1975. It's the International Year of the Ivory Trade Ban. But you have some difficulties in the start of this material because, as we know, we have to date the parts that stop changing carbon with the environment. So you have to date the parts near to the skull of the elephant. But in this work, you are in work with modified ivory piece, so which can be extracted from any part of the tusk. So you have pieces can be from the I don't know. There is the point. For the tip, for the middle, or for the near of the school of the material. You have here some kind of netzug. There is this one here, and another one there. And these materials is a small ivory figure, so we don't know which part of the tusk is from this material. You have earrings, you have necklace and bracelets. This necklace here, it's a good example because you have some bows. And one bow can be for the one tusk, another bow can be another tusk or another elephant. So it's really important to date a minimum of two sampling of the sample to compare the results. So we use the Schweger partner. This is a partner commonly used in forensic cases about ivory to separate the ivory from mammoth or ivory from elephant. The difference is the angle of this partner. So for mammoth, the angle is until 80 degrees, and for elephant is more than 100 degrees. And in this work from Alberic et al., she's talking about the mineralization of collagen fibers. So this is important to us because we have to date the parts that contain collagen in the sample. So here you have the partner in 3D here and 3D format. In this work, Alberic talking about the aerial plane is the plane that contains more concentration of collagen. So here there is a video. You can see how the partner is in the tusk. So here the partner. So we are looking for the aerial plane. The aerial plane is this partner here. This is the. Okay, great. This partner here from the tusk that contains more concentration of collagen. So now I'm going to talk about the protocol used for these samples. We have 3 steps. We start with the calcification process. We use the acid 0.5 molar for 18 hours at room temperature. And the next step, use the ABA, the acid-base acid, to do the decontamination of the process. So we use the base from 0.1 molar for 30 minutes at room temperature. And the next is the acid again, 0.5 molar for an hour at room temperature. And the last step is more important because in this step you're going to know if there are collagen in your samples because the gelatinization process. So use the pH 3 for 20 hours at 70 Celsius degree. And the next step is, because of that, is modified from the logging here because the next step is the ultrafiltration step. And they use this when the sample is not there is no good conservation. So our sample is modern, so we skip this part and use the freezing dry for 8 hours. Here you have the calibration curve. This is a work from Hua et al. This is important to us to know where your samples come from, and they use this curve to calibration. So you have some curves for North Hemisphere and South Hemisphere, and your sample specifically is from the Central Africa. So you're going to use the curves for North Hemisphere Zone 3 and South Hemisphere Zone 3 to do a calibration. Now, talking about results, we conduct a total of 3 tests. The first two were preliminary tests, and with the crucial difference being sampling methods. So in the first test, sampling was conducted with the aim of study of the spiral growth of the tusk from the inside out. This spiral is from the partner, the growth. And in the second, you attempt to sample according to the growth sequence. And in the third, we utilize the Schwager partner, specifically looking for the error plane, the plane that contains more collagen concentration. Now, when I'm talking about some results of our work, it's important to know this is the sampling in pairs. So AB, it's one pair, AF is another pair, IJ another pair, KL another pair. You have 3 tests, and I wanted to talking about some things here. About the PMC, you can see how the PMC is more than 100. That means how samples is modern. And about the calibrated age, we have here how the results. There is a probability be after 1975 is the year of the law. And about the mass, here in the test 1, you have the bulk mass 43, 49 milligrams, and the college mass was 2-1. It's not so good for us because it was looking for a mass for about 5 milligrams. But in the second test and the third test, you have a good college mass because we did the sampling in the parts that contain more collagen in the aerial plane for test 2, and the test 3, you did the sampling in the airplane or near this plane, so you have a good collagen mass. Now here we have some location for S samples for bracelets for the sampling A and B. You did the sampling inner and out, and the sampling location for the pair AF. It is the sample lower and the upper. And now, talking about the results you have, here you analyze the part that's the probability of the sample being post-1975 according to the law. So we use both curves for North Hemisphere and South Hemisphere. Here, the results from the bracelets. So you analyze like one sample for both curves. So we want the percentage, the probability to be above 95%. So both here can't be commercialized according to these results. And in the figure, you have the results for how probability of these samples, this pair. And about the second test, you did the sampling in the upper and the below, and both have the results. The probability above 95%, so it's good. And here is interesting because the agreements of the samples are really good because you did the sample upper and the lower, so you imagine that the results will be really close. So it's really easy. About the test, the third test, you did it for a ring. This is interesting because a ring 1 and ring 2, we analyze separately because a ring 1 can be from the one tusk and ring to another tusk or another elephant, so analysis will be different. And here you have the percentage below 95%, so this can't be this can be commercialized according to the law, but this another result. It's a kind of netsuke. I told you before, there's small carved ivory figures, so you did the sample upper and the lower too, and both is above 95%. And here is the probability for how the samples, I forgot to say, in green you have the results for South Hemisphere, and in black, only here you can see. We have the results for North Hemisphere. Now, about the conclusion, this study enables us to sample and date modify ivory samples, and through the process, the growth pattern of tusk was observed and used it to modeling a chronological sequence. And this approach offers a valuable tool in preventing the illegal trade of ivory and the other associated criminal activities. And about some future perspective, we will know in my PhD, is conduct a study of tusk growth, taking into account the influence of seasonality and assess the potential contaminants that could be used to falsify the age of the ivory, and consider the time lag in the sample, the interval between the incorporation of the carbon into the ivory and its measurement. And the second goal is because we are treated with the forensic cases, so someone can put something in the samples to age be more older than they really are. So thank you.
You want to sing something? No. No. Okay. So we have some samba. So yeah, we have a question. John, Tim, please.
So it's a very nice talk, but to be sure that if you calculate 95% after 1975, is that enough for the police to convict the smuggler? Because if he has a fancy lawyer, he will say there's still 5% over there.
Yeah, but the results about the presentation, we want to be, how can I say? This, the trade is legal, but the age only to be commercialized or not if you can keep this type of sample.
Okay, Juan?
No. Oh, so the second question is a legal question. So the CITES is legal in the year that the country recognized the treaty. So is that in 1975 in Brazil, or is it later? Do you know? Maybe you don't know the answer to that.
I don't need to. Sorry.
So you can, it is illegal from the moment the country recognized this treaty. So if, for example, Brazil recognized in 95, whatever it's older, it's okay. It can be ivory from 94, and it's not illegal.
Yes, according to ivory, according to the states, yeah.
Okay, Juan, and then Walter.
Okay, just one comment about the calibration.
Yeah.
Always because the bomb curve, the rising, they have a less data point than the falling part. Then the upscale or other calibration curve usually give you more, higher population after the rising of the bomb. And it's very difficult to say exactly, but if you have a series of data, then that can be improved. But for a single day, be careful with the probability based on the calibration curve, because always lower probability for the rising compared to the falling. Okay, just my comment.
No? No? When it's going from the 1955 to 1963, there is only a few points, but from the 1963 down, there is every year point. The rise, it's imprecise. We cannot say what happened within 1 year, because we have only one average value. Behind there is much more precise.
Oh, yeah.
Okay. Yeah, so any other question? Walter, you want to?
I don't know where the samples came from, but I know that for a fact that the very big illegal trafficking with ivories in the Far East, and these are African elephants, and there are big papers showing how tons and tons of ivory came from elephants killed after the ban. This is quite easy to actually show that. So I'm not sure whether in Brazil there's really a big problem of that. As far as I know, in the world distribution, the big market is in the Far East. That's for sure. And unfortunately, it's mainly African elephants which are being killed far after the ban was raised. By the way, the 1975 was the original agreement, I think, at the UN, but eventually each country has to approve this date. And so the latest were even in the 80s or early 90s. So many countries joined this ban now, but at different times. Yeah. So for each country, when you find something, it's important that you show that this ivory was actually from elephants killed before that ban, this specific ban in the country. So this is a, but as Tim points out, the lawyers always find out what is the real, when you actually have to get into consequences of such traffic. Okay, sorry.
Okay, one last question from Matthias.
I'm wondering, thanks for the talk. I'm wondering, since the title in the talk mentioned the Schrager patterns, I wonder how you use them, because it is very essential to know when the last part of the ivory is grown. And in jewelries, you do have a big problem not knowing where it is coming from. So the Schrager patterns essentially could be useful, and I wonder how you do that.
Okay, so it's a jewelry. It's not the whole piece of the task. So how do you know that what you are measuring is the last growing age? If you know the jewelry is not from the part which is from the middle and not from the tip of the task.
Yeah, you don't know which department is from the object. Because of this, you use the department to date the part that contain more collage concentration. Yeah. If I can know which department from is the bracelet, for example, in the Tursday.
Let's say by now.
We don't know which the exactly part is the object where they.
Yeah, I think now it works only that whenever it's younger, it's prohibited.
Okay, later.
Okay, so we can thank you for the wonderful talk. Let's thank all of the speakers. And now I can close the first session. I'm so sad because it was nice, I would say. Thank you. See you on the coffee.
So before start the second session today, just an information also for today and for tonight. I hope that everybody can hear and can be here. So today we have the poster session 2 from 16:30 to 17:30. So please remove the previous posters if you have not done yet, because they have to put the new one. Just at the end of the poster session, there will be 2 buses, as yesterday, that will leave to Cedad. So we have to be on the bus, you have to be on the 2 buses at most 17:35, because we have to move from Lecce to Brindisi and Cedad. Cedad is it's there. And then we have to visit the laboratory. It's not easy to manage more than 100 people at the same time in a short time, so you will be quite free making questions if you want. There will be also the second floor of the CEDAD if you want to have a transparent drink, very light, because we have the gala dinner later. At 19:15, from CEDAD, we will move to Castello Monaci for the gala dinner. Okay? Try to be punctual. If you have, I don't know, to wash, to leave, to change your clothes before going to CEDAD, there will be no possibility from Lecce from CEDAD to go back to Lecce before going to the gala dinner. So from CEDAD, we will go to gala dinner, Castello Monaci directly. So you have plenty of time, if you have the bed and breakfast here or the hotel here, to do whatever you like. On the badge, I didn't be more clear. So we are in the Salento. This is a dialect. Salento, l'usule, l'umare, l'uiento means the sun, the sea, and the wind. You will find everywhere, even on the shirts or everywhere. Also, on the back of your badge, there is oru oru ognetuno a casa loro. It's a dialect from this part of Italy. That means when we finish the session, okay, oru oru, that means we go home. Fast, we have finished, we go home. Oru oru, you will hear also this. Pizzica, you know what is the pizzica. Be ready for this night. And also I upgraded the leitmotifs of the conference because it changed during the days. Because there is an important, Golden Ugla Award that will be for tomorrow. So we started with volare o cantare o, okay? All the other attendees or participants that sang, for instance, Va pensiero or what Figaro will be eliminated because you got only two choices, volare o vincero the Nessun Dorma. So we started with volare and cantare. And we changed it during the conference, volare o, danzare o, that means dancing. And then also volare o, suonare o, to play. And we have already seen, also yesterday, and we'll see this evening, volare o, mangiare, that means to eat in Italian. And yesterday, volare o, nuotare. On the beach. Maybe some of you will recognize himself there. Okay, enjoy the session, and the chairman knows that he has to respect the time and the schedule of the conference for all these reasons. Okay, thank you.
Okay, thank you. Okay, we start the second section of A1. The first talk is Philippa Ashcroft. She will talk about the residence time stability of soil organic carbon through RAM oxidation and the carbon-14 analysis. Yeah.
Thank you. I don't know, should I stand on the box? Stand? Do I stand?
If you want.
Okay. Right, I'll try this. Can you hear me? Just nodding. Okay, thank you very much for coming to my talk, and thank you very much for the organisers. Really happy to be here, especially because I checked the weather back home this morning and it's 11 degrees, raining with wind in East Kilbride, so I might not show my lab members the photos of us on the beach yesterday because they might be a bit too jealous. So, this is a presentation about some work that we've been doing using ramped oxidation and C14 analysis. And as we've seen, there's been quite a lot of interest in ramped oxidation and ramped pyrolysis oxidation in the community over the past few years with units developed. And we saw some interesting presentations and papers coming out. And then in about 2018, 2019, we decided that we would construct our own system because we were interested particularly to look at soils. We do quite a lot of work on soils and sediments in our lab, and we have quite a pedigree of looking at different soil fractions. We've looked at pyrogenic carbon and different techniques for sort of separating different fractions of soil out. So we're really interested to see how this technique might assist us. So this presentation is actually not going to focus on the technique particularly itself that we've got. It's more on the first sort of real application that we've been using it for in a consistent way. So I'm going to give you a bit of a background as to this application and this project and the use of the ramped oxidation. So this is a project that we're involved in looking at the transition in the UK from temperate grassland in the top picture to coniferous forests. And in the UK, as in a lot of other countries at the moment, there's quite a lot of movement towards things like carbon credits, and lots of companies offering carbon offsetting through afforestation, planting of trees. Which is it sounds very simple. You plant the trees, you grow the carbon, you offset what you've emitted because you're storing this carbon. But like anything, it's never as simple as it sounds. So there's work to be done to kind of figure out what it actually means when you start to take this action, changing land for carbon credits. In the UK, we have this really big history of conversion of temperate grasslands to forests. This really started in the 20th century, so the First World War, the Second World War. There was a movement in the UK to become not dependent on foreign sources of timber. So there was all this movement for fast-growing coniferous forests. And then later on, there were financial incentives given to landowners to convert land to forestry. The forestry industry in the UK is significant for us, but not massive worldwide. We still import a lot of our timber. But there's quite a lot of this has happened. There's also climate warming, so the tree lines are starting to move up. So some of these temperate grasslands are starting to convert to forest land. So usually up until about the 1990s, it was fast-growing coniferous forests that we used. So if we look at those two pictures, they look very different. So if you walked through those two environments, even with your eyes shut, you would be able to smell differences, it would feel different. However, they're also very different below ground. And the key thing for this project is looking at mycorrhizal fungi associations with the plants. So about 80% of terrestrial land plants are dependent on some sort of an association with mycorrhizal fungi in the soil. I won't spend too much time on this. I have a timer ticking away here. So essentially, in a grassland system, we're tending towards this arbuscular mycorrhizal association. So these are particular type of fungi species that they penetrate the cell root in the roots of the plant. So they're actually in the plant, and the plant's in them. Why plants want to associate or have evolved to associate with fungi is a mutualistic relationship. So the fungi provide extra moisture and nutrients to the plants. However, the fungi are below ground, so they can't photosynthesize. So the plants give the fungi up to about 20% of the carbon that the plant has fixed. So there's this system here. In a coniferous forest, so ectomycorrhizal fungi associations are what's happening. So these are fungi that are outside the cells of the plant roots, so the ecto, kind of the clues in the name there. And these different fungal associations create differences for cycling of litter, but also cycling of carbon within the soil systems. So if I move on, so the picture here, this is a really nice study by Wu et al. In 2022. I don't have a pointer, but you can see which image is the graph, hopefully, on that one. So they did a massive meta-analysis of like thousands of publications looking for fungal associations. And what they found was that in an ectomycorrhizal fungi system, so like a forest soil system, what's happening is the carbon storage, there's more carbon storage, but it's tending more towards the particulate organic matter, so the POM, and that's a word that's going to be important for the rest of the talk. So the POM is poorly degraded litter material, perhaps less degraded plant material, animal material, that sort of thing. And it's thought to be faster turnover. In an arbuscular mycorrhizal system, you can see here on the right that the carbon is more moved towards this mineral-associated organic matter, or MAOM. So that is carbon that's stuck to soil minerals. It's thought to be, by convention, more resistant, slower turnover, and hosting kind of a longer carbon storage. So the basis for this project was looking at, if you're changing the vegetation, you will change the fungi. That's non-negotiable. But what does it do to change the soil carbon storage? So that led to this project, Mycorrhizal Drivers of Soil Organic Matter Formation. And why we would want to do this is because for policymakers and land managers, it is quite important to understand if you're afforesting, you're putting carbon in the biomass above ground. But if what you're doing is changing the cycling of carbon below ground, and you're releasing carbon more quickly in that system, it actually then becomes a zero-sum game. So any carbon benefits might be offset or even negated by other things that you've not considered. So for this study, 4 sites in Scotland. These are sites that have been converted to grassland about 60 years ago, and there's information for how that sequence has developed. And at those sites, they're important because there's an equivalent site that's remained as temperate grassland that's in the same climatic zone, the same local area, and it's a good comparison as a control, and where the 2 fungal associations, their impacts can be compared. So OL is forest, and GR is grassland in the rest of the talk. So the bulk soil is the 0 to 5 centimetre soil layer that's been used for the data that I'm going to be presenting. So the first stage was actually just looking at the bulk soil analysis as we would do with our standard techniques. And the hypothesis was that there would be a statistically significant difference between the C14 and the carbon storage time in these 2 systems. And although on the right-hand side it would look by eyeballing the graph that there is a difference, we need to look at this statistically. And there was no consistent statistically significant difference between the 2 systems. So that then becomes a bit of a question of what's going on. Is there no treatment effect whatsoever through this conversion, which is one possibility, or are we just not picking it up with the methods that we've got? So the keen-eyed will have noticed that it's probably quite unlikely that we've got 3,600 radiocarbon-year-old organic matter in the topsoil. Later on, some of the people involved in the project said, oh, this is near to some old coal mining sites, which was information that was probably useful earlier. However, there is a slight silver lining to that later on. So this is, I'll go through this quickly in terms of time, but really on the left-hand side, just to remind you, particulate organic matter, the POM, fast cycling, fast turnover, and it's coming more from the litter itself, and the mineral-associated organic matter, MAOM, on the right. So if you have bulk soil, you can do the C14 on the bulk soil and compare, but you can also fractionate. There's many schemes to do this, but they're usually some form of chemical separation like sieving or a density separation to get the POM, the MAOM, out. So this was actually something that was then investigated after the bulk soil showed no differences. However, again, the radiocarbon results after quite a laborious fractionation that I didn't do myself. There was a very keen postdoc who was less keen probably by the end of several months doing this. But, you know, he didn't find any statistically significant difference. So there's either something really complicated that we can draw out of this and kind of fit the explanation to the data, or again, we're still not really picking anything up with this method. So that then led us to think, right, well, perhaps what we need is almost a kind of chemical microscope approach here to go deeper into the chemistry of the different fractions that are represented in the bulk soil. So I'm just going with the bulk soil in the rest of the talk. And using a thermal separation method, it's something that's more, it's on a chemically defined basis because it's the activation energy of the molecules that are in those mixtures are something that's kind of reproducible chemically. I'm not saying that each soil that we sampled has the same chemistry, but there'll be thermal fractions. Those might be different inside of themselves, but it's something different than the more conventional physical separation, which is on an operationally defined basis. So we went to have a look at that. This is our system. I'm sure you all studied our poster in minute detail on Tuesday. If anyone is sad because they missed it, I'm really happy to chat over a drink tonight. And we have a paper out last year. So the thermograms, firstly, of the bulk soils on the left show, surprisingly, from all of these different sites, they're not particularly different. We find that thermograms that look quite similar from similar locations probably show a similar chemistry. You can compare that to different organic fractions that we're working on in other projects on the right. So a coastal salt marsh sediment, the big peak at the end is probably carbonates and peats and things like that. So we've got soils that are quite similar. That's then normalized to the percent of the peak concentration. This site 2, so these are just sites 1 and 2. Site 2 was the one that was near the coal mines and had the really low radiocarbon ages. Potentially there's something at higher temperature there that is the polyaromatic fraction of the coal that we're seeing. So in terms of what to actually analyze, we decided to use a peak deconvolution approach here. Because of the similarity of the different thermograms from the different soils, it indicates it's similar sorts of groups of material. We don't know for sure. So we decided to write a Python program to then look at these peaks and figure out what's the best fit for the lowest number of peaks that we can fit in, so as to just make things less complicated for ourselves in a first pass at looking at this. Obviously, we could do other things as we go through this study. We could separate by temperature intervals and looking by eye. But this is what we elected to do. So that gave us these 3 temperature intervals that we decided to use reproducibly for every soil sample on the left-hand side. You can see that's the high temperature fraction from site 2 with the coal is highlighted. On the right-hand side is anthracite coal from southern Scotland. So temperature-wise, the one sort of positive from having one of the sites affected by coal is there might be some potential for using this technique to try and at least improve dating when you have a really problematic soil that's got some coal in it. That really even things like high pi wouldn't get that out. So leaving the coal aside, because it's not really the main thrust of what we're looking at here, so the rest of the data we're ignoring site 2. So the question of does the C14 content, or the PMC, vary with the temperature in the ramped oxidation? The answer is yes. Statistically, using one-way ANOVA, there's a significant temperature effect. This is something that's consistent with previous observations from other groups that have looked at soil. For example, I know Susan Trumbore with Shane Stoner worked with the ETH and found a similar outcome, that the higher temperature fractions were the older fractions in soil ramped oxidation. So then the second question of, is treatment effects now observable? The answer is yes. So the polynomial curves are just to kind of aid the eye in case anybody didn't have enough coffee. I'm not suggesting that there's a polynomial relationship really here. The effect is largest at the low temperature, but really what we're seeing is a higher PMC now consistently in the grassland sites, the forest sites, as compared to the grassland sites. When we recombine all of these fractions, we get a similar result on the right as a weighted average to what we got when we did the first analysis of just the bulk soils. So a bulk C14 measurement, even if we combined all these fractions, wouldn't statistically identify a treatment effect. So what does this all mean? So it's important in this to remember that the samples are potentially a mix of pre- and post-bomb carbon. So we have to bear that in mind, that not all of these samples might be kind of in the same direction. However, we do feel that we're getting something, a consistent message out of these, and that there is more work to do. So the answers to kind of the overall graph on the right, where the reason for this might be that there's a slower turnover in the forest soils, so the litter is decaying more slowly, you've got more woody litter, higher percent carbon, and that's got more bomb carbon entrained in it. There's also a question that perhaps this is not to do with that, it's to do with a higher retention of old pre-bomb carbon in the grassland soils. One answer potentially is that it's both things at the same time. So if you go right back to the Wu paper, if you remember, the arbuscular systems moving carbon into more of the MAOM, the fraction, would be consistent with retaining older carbon in that fraction. And the ectomycorrhizal associations moving carbon into the POM and the litter, that would be consistent with what we see here at the same time. However, I'm not going to sort of stand here and say we've cracked this answer definitively. We're working with colleagues because we need bulk density measurements to look at the actual carbon distribution in these soils, and we're talking to modelers to look at carbon turnover. So next steps, we're going to start looking with the same approach, the same fraction, the temperature windows on the different fractions. We've got the thermograms of those. FLF is fine light fraction here, so even lighter than the POM fraction. And we can see some differences starting to emerge in these sites when we actually run the ramped oxidation of the different fractions. But we need to do the C14 of the different windows, temperature windows, in order to see are we going to get a more fine detail picture. So I'll put the conclusions up there. I can see 1 minute 36, so I'll let you read the conclusions yourselves. But the important thing here is that without something like a ramped oxidation approach, we wouldn't really have been able to pick up in these sites that a change in landscape use and change in vegetation was really affecting the carbon cycle at a fine detail level. So, yeah, thank you very much for your attention. I have a voice like I think it was compared to a strangled cat once, so if anyone else wants to sing, feel free.
Okay, I have a quick question. Time for a quick question.
What actually did you analyze? What soil horizon and how depth your samples?
So notes to...
what horizon?
What horizon? The top horizon.
Were analyzed?
So it's the top part of the A horizon.
So top 10?
Yes, centimeters. So remove the vegetation, and this is the active horizon, which we're most interested in for soil carbon storage.
Yeah, because the forest affect, they change the root system from the surface in a grassland and much deeper.
Yeah, so the forest, they still have it's the surface roots, and every root will have, everywhere, will have a mycorrhizal fungi association. So there are studies showing with depth that the carbon storage and the carbon turnover actually starts to vary. The Wu et al. Paper has quite a nice summary of how that works. So in this study, we've just looked so far at the top soil horizons, but one would ideally want to go in slices with a depth profile. Yeah.
Okay, thank you.
Sorry, I have been told I have to be strict with time. You can talk to. Sorry about it. Okay. Can we move to? Thanks, Philippa. Okay. The next presentation is Husser's. He will talk about the molecular level of soil carbon turnover and the amino acid specific carbon 14. Yeah.
Yes.
Welcome everyone, and thank you for joining my presentation on molecular soil level soil carbon turnover unveiled with amino acid specific radiocarbon. To set the stage, radiocarbon is fixed by plants via photosynthesis and later assimilated by animals and microbes via the food web. After the death of an organism, there is no longer an exchange with the surrounding environment, and the radiocarbon stock slowly depletes. In soils, the nutritional value of organic matter depends on many factors, such as the type of molecules available, their energy content, and their accessibility, meaning how easily these molecules can be broken down and utilized by an organism. We generally have two different pools, as we referred in the last talk. Well, there are many more pools, but for simplicity reasons, we say we have two pools. This POM pool and the mineral associated, the organic matter pool. Yes. So POM is generally more accessible and has fast turnover rates, while MOM consists of organic matter that interacts with minerals, leading to stabilization and a slower turnover. As a simplified concept, POM with a fast turnover follows the atmospheric bomb curve quite well, which is shown here with the green line, while the slower turnover of MOM leads to a slow and more persistent increase in radiocarbon, shown by the red line. Bulk soil, as a mixture of both POM and MOM, is somewhere in between. On a molecular level, soil consists of a complex mixture of compounds with different behavior. Some of these compounds are also providing information on their source and are therefore used as so-called biomarkers. For example, N-alkynes, long-chain fatty acids, and lignin phenols are plant biomarkers, whereas short-chain fatty acids are biomarker for living, and GDGTs for diseased microbial biomatter. We also have molecules that are ubiquitous in all organisms, such as amino acids or nucleic acids. And in this presentation, I will focus on amino acids. So amino acids have a high potential to interact with minerals due to their carboxylic acid and amine groups, which can form hydrogen bonds with mineral surfaces. They are ubiquitous in all organisms as the building blocks of proteins, peptides, and enzymes, and serve as a valuable nitrogen source. In soils, proteins and amino acids make around 40% of total organic nitrogen and contribute significantly to the total organic carbon pool, playing a crucial role in soil health. There are 22 different amino acids in proteins, which are listed on the right. These amino acids vary by their side chain, which determines their properties. Despite the vast abundance, individual amino acids have not yet been the focus of radiocarbon analysis. To address this, we developed and tested a method to measure radiocarbon in individual amino acids. We applied this method to soil samples collected from two well-studied sites in Switzerland, Beatenberg and Lausanne. These sites feature diverse soil types, podzol in the subalpine region of Beatenberg, and cambisol on the Swiss plateau at Lausanne, offering a solid foundation for our analysis. Both locations have been extensively studied as part of the long-term forest observation program of the WSL, the Swiss Federal Institute for Forest, Snow and Landscape Research, making them ideal for testing the radiocarbon content of individual amino acids under different environmental conditions. We collected soil samples from various depths at each site, allowing us to investigate how soil amino acids behave and age across different depths and soil types. Previous biomarker studies have already provided valuable insights into the behavior of fatty acid, N-alkanes, and GDGTs. To extract and isolate amino acids from these soils, we first demineralize the samples using hydrofluoric acid to release the amino acids and proteins bound to minerals. Next, the protein and peptides were hydrolyzed, followed by a desalting step to reduce the salt content. This process resulted in an amino acid solution that could be separated and isolated using high-performance liquid chromatography, or HPLC. HPLC is a liquid chromatographic technique that pushes a sample through a column under high pressure, allowing it to interact with the column material and separate into individual compounds. Most columns are packed with porous silica with modified surface groups that allowed for tailored solution for different applications. Typically, the carbon content in silica columns ranges from 3 to 20%. However, a major drawback is that silica-based columns require specific pH and temperature conditions and are prone to degradation and column bleeding. For the separation of our amino acids, we opted for a porous graphitic carbon, or PGC, column instead. This column has a carbon content of 100%, which might seem concerning for radiocarbon analysis, but rest assured it's not. Graphitic carbon is highly stable, and columns packed with this material can be used at any pH and at temperatures up to 200 degrees Celsius, all while maintaining an extremely low column bleed. PGC columns are particularly effective for separating very polar molecules like amino acids. However, they are quite expensive. With a semi-preparative PGC column, we developed and validated a method for radiocarbon analysis of amino acids. The chromatography is faster than previous methods, taking less than 2 hours per run, and does not require modification of the amino acids, avoiding extraneous carbon. The retention times were consistent across different injection amounts, and radiocarbon measurements on amino acid standards showed that we have a stable blank of around 1 microgram of carbon with a fraction margin of 0.6 and no column bleed. On the left, you can see the chromatogram of a standard amino acid mixture. Now, let's look at our soil samples. Although they appear more complex with additional peaks, it's clear that amino acids are present. Especially at lower soil depths, the presence of salt is also clearly visible. Looking at the concentrations, we observed that absolute values decreased with increasing soil depth at both sites, and additionally we found higher concentrations in Battenberg, especially at this top soil layer compared to Lausanne. When looking at organic carbon normalized amino acid concentrations, so concentrations increase with depth and constitute a significant fraction of organic carbon. In Lausanne, amino acids make about 3 to 5% of soil organic carbon, with maximum concentrations at 10 to 20 centimeters soil depth. In Beatenberg, concentrations steadily increase with depth, reaching around 2 to 3%. At both sites, histidine shows a notable increase with depth. This basic amino acid has a higher potential for mineral surface interaction. When we look at the radiocarbon in our amino acids, the delta 14C values decrease with depth, as expected. Notably, histidine shows the lowest delta 14C values, and which is supporting the hypothesis of mineral stabilization. Please note that for Lausanne, the deepest soil sample is missing due to a very low TOC content of only about 0.2%. And for clarity, amino acids are highlighted in red in the following graphs. When comparing amino acids to soil, bulk soil, density fractions, and DOC, we observed that some distinct patterns. In Beatenberg, amino acids show higher delta 14C values than bulk soil, but slightly lower than DOC, except at 20 to 40 centimeter depth, where POM is higher. In Lausanne, the topsoil shows lower delta 14C values for amino acids compared to bulk soil, while at 10 to 20 centimeter depth, the values are slightly higher. Further comparison with other biomarkers that shows that in Lausanne, amino acids have lower values than plant and living microbial biomarkers, but higher than disease microbial biomarkers in the topsoil. In the deeper soil, amino acids exceed other biomarkers. For Beatenberg, amino acids consistently show higher values than other biomarkers, except for the living microbial biomarkers at the lowest depth. When we plot compound delta 14C against bulk delta 14C, biomarkers above the 1-to-1 line generally indicate faster turnover, while those below suggest slower turnover. However, recent atmospheric radiocarbon levels nearing and dropping below 0 per mil add complexity to this interpretation. Going back to the plot from the introduction, when we look at around 1980, we see that the slow turnover radiocarbon signal actually surpasses the fast turnover signal. In Lausanne, bulk delta 14C is slightly higher than amino acid delta 14C, indicating that amino acids reflect more of the recent atmospheric radiocarbon signal. At Beatenberg, despite the slow cycling environment of the pot soil with low biological activity and high recalcitrant organic matter, amino acids exhibit a faster turnover than bulk soil, demonstrating their dynamic nature even under stable conditions. Notably, at greater depth, amino acids tend to align more closely with bulk delta 14C behavior, reflecting a similar turnover rate and mineral interactions. Now let's move to a more playful representation. In this graph, I've plotted the delta 14C of the amino acids onto the atmospheric bump curve. So this visualization shows that amino acids are relatively modern, with the oldest one being just about 17 years old. And it's a lighthearted way to highlight just how dynamic and recent these compounds can be, even in environments where there might be a slower turnover expected. So in conclusion, we have successfully developed a method for conducting radiocarbon analysis of individual amino acids in soils, but not only soils. This method is also applicable to basically any other material containing proteins and amino acids, which this allows us to gain a more detailed understanding of soil carbon dynamics. And we found that amino acids represent a significant and active pool of both carbon and nitrogen in soils, playing a crucial role in nutrient cycling. Our findings show that amino acids have a fast turnover, reflecting their dynamic nature within the ecosystem. Lastly, we observed that turnover rates are strongly influenced by soil type and depth, highlighting the importance of these factors in controlling amino acids dynamics in different environments. Yes, thank you.
Yeah.
Are you going to sing? You sing. No. Okay, I had a question. What is the food for microbiome? Because sometimes you show the microbiome value is higher than the plant wax, but sometimes it's lower than the plant wax. It's a different depth. What the food? Yeah, what the food for the microbiome?
The food is, I think it's there are many sources. For sure, at higher or at lower soil depths, I think is leaf litter and essentially any other accessible organic material.
Okay, time for a couple of questions. Yeah.
Dating amino acids individual is an important part for bone dating. Is there any correlation or any exchange of experience with dating amino acids in soil and also in bone?
Yes, so in bones, the advantage for bones is that you can target hydroxyproline, which is an animal-derived amino acid. I've had some exchanges with people doing this, but our approach is completely new, I would say.
Okay.
Any more questions? Okay. If not, we move to the next, presenter. Thank you. Okay, the next presenter is the Carla Haddon. She will talk about the age offset in the postponed terrestrial scenario from South Florida.
All right, thank you for that introduction. My name is Carla Haddon. I'm from the University of Georgia. And first, I wanted to say congratulations and thank you to the conference organizers. This has been a really well put together conference. I'm really enjoying it. And I especially enjoyed the field trips yesterday. That was an awful lot of fun. So now I'm going to tell you a little story about some snails. I will get to the terrestrial snails from Florida, but first I have to explain a little bit about why I'm interested in these snails in the first place. And so the story starts in the coastal area, in the marine area. So from an archaeological perspective, shell-midden sites in the southeast U.S. are some of the most important archaeological site types in the region. They range from kind of small midden scatters to really large circular depositions, these shell rings, and these represent some of the earliest permanent villages in the southeast United States. To date, there have been about 4,000 that have been documented, but that is a kind of an underestimate because over the past few hundred years, a lot of these have been lost to development. Also because there are such dense concentrations of shell, over the past 200 years, they've been really aggressively mined for building roads and things like that, and for a number of things, building tabby houses and whatnot. So what's left of them is a really important archive of human history in the southeast U.S. Unfortunately, the ones that are remaining are still very vulnerable. These are at the very edge of the sea, and so with rising sea level, they're at risk of inundation and loss. And also with more frequency of hurricanes and more intense hurricanes, a lot of these are being really destroyed very quickly. So here's an example of a site I worked in. It's not showing. The middle one is the site I worked in the Everglades. This was right after Hurricane Irma. So it just completely cut this giant mound in half. So one more big hurricane and it's probably going to be gone. And in fact, there is a big hurricane heading towards Florida right now as we speak. Hurricane Helene is expected to make landfall tonight. And it's so big and so bad that they've actually closed the University of Georgia campus, even though it's hundreds of miles away. So these sites are really at risk of being lost, and some of the early sites are already underwater. So there's kind of an urgency to study these sites as much and as completely as possible. So over the past 10 or 20 years, there's been a tendency for archaeologists to kind of prioritize quantity of dates over quality of dates. the most abundant material for dating is shell in these shell midden sites. So some big survey programs have really focused on dating things like oyster shell, marine snails, and to a lesser extent charcoal when it's easy to find, but these are really shell-dominated sites. So a lot of these chronologies have been based on shell, which of course is problematic for the reasons that Susan was telling us about yesterday. So for the past 10 years or so, I've been working with archaeologists to try to help better understand the range of variability in marine reservoir effects and shells in this region, and maybe identify some shells that would be better or worse for dating moving forward. Okay. So as we all know, marine reservoir effect, the effect of that is that marine shell dates tend to be older than contemporary terrestrial materials. So I started my work in marine reservoir effects focusing on species that were most abundant in archaeological shell sites in the southeast. Some came from truly marine environments, so these big conch shells, lightning whelks, and things like that, but then also oyster shells, which are common in the estuaries. Moving on here. They all have problems, and as Susan pointed out, you have to really pay attention to the species, the local environment, the particular habits, ecologies, ecosystems. These are really specific details that you have to know about your system that you're working in to have a really robust delta R correction. So this is kind of a mass accumulation of all the delta R data I've been working on for the west coast of Florida, and there's a really big range. So these offsets can be anywhere from -200 to +200 or more extreme. Even within the life of a single individual organism, you see these major fluctuations in the scale of this reservoir offset. So for a precise chronology, that's not very useful. So when we're talking about shell-based chronologies, we're really painting with broad strokes here. So you can tell Pleistocene from Holocene, no problem, but if you're looking for like generational scale human histories, you can't do that with marine shell dates. So I've been working with archaeologists to try to think about what's a better way to get at chronologies, looking at these really shell-dense sites. Is there a different species that we could be thinking about or looking at? So the next critter I went to, and I really love this animal, this is the marsh periwinkle, and it's kind of semi-terrestrial. It lives right at the water's edge, and it lives on the marsh grasses, and it moves up and down. It's got a daily vertical migration. It eats the terrestrial plants. So I was hoping that maybe we would get a terrestrial date from these critters. So a few years ago, I did some experiments. The shell carbonate looks just like marine DIC. So forget it, you can't do the shell carbonate, it's no better than oyster, for example. But the soft tissue looked just like the modern plants, so that was really promising. Of course, the soft tissue doesn't preserve archaeologically, but there's a trace amount of organic material that is part of the shell, the shell itself. So it's this very minute kind of organic matrix that helps the biomineralization, kind of directs biomineralization, kind of broadly call it conchiolin. So I was able to isolate conchiolin and date that, and that gave us a really good date, a good terrestrial date. But the problem is that the sample masses were so tiny, anything that you gained by targeting that trace amount of conchiolin, you lost in terms of precision because the samples were just so tiny. So that was kind of a dead end. So now my archaeologist friends are really interested in these truly terrestrial snails, these little garden snails we call them. These are critters that people weren't collecting them and they weren't eating them, but they're really common in these shell sites for a couple of different reasons. So my friends were wondering, is this something that we can date to get a reliable date that's at least related to the time of deposition? So for me, the question is, are these shells putting carbon into, are these animals putting carbon into their shells in equilibrium with the atmosphere? So to answer that question, I figured the best way to do it was to look at shells that are of precisely known collection date from the post-bomb period. If you dated those shells, do they land on the bomb curve or not? So that's what we did. So these two species in particular, we're focusing on them because they're terrestrial, but they're really common in archaeological sites. You can grab any handful of shell midden and you're going to get one of these two critters in that little grab sample. They have pretty short lifespans. It's kind of speculative, but maybe a couple years at most. And they're really easy to identify. You don't have to be a specialist. You can tell very easily, is this a rosy wolf snail or is this a flat coil? So this seemed like a really easy way to go. They have slightly different, very different habits. So the little flat coils, they like eating kind of decaying vegetation, and so they're really attracted to human garbage. So archaeologists call them commensal. They're really common in middens because they're attracted to the garbage. The rosy wolf snail, and I love that name by the way, they're wolf snails because they're carnivorous. So they're not very big, they're maybe the size of my thumb, but the wolf snails are attracted to the flat coils, so you'll often find them in the same assemblages. The question that we really needed to get to with the land snails is where is their carbon coming from? So there's kind of 3 general pathways where they could be getting carbon for their shells. Certainly it could be coming from CO2 in the atmosphere. It gets dissolved in water that's on the ground and dew and rain droplets, and that could be incorporated into their shells. And that would be a good thing. They could get it through diet. So CO2 is absorbed by the plants. They eat the decaying plants, and then it works its way into their shells. And that also would be a good thing. But the concern is that they could also incorporate really, really old carbon if there's limestone in the local environment. This is kind of referred to as the limestone problem, and it really is quite variable regionally and also from one species to another, whether or not they will incorporate limestone if it's present. So we wanted to see, are we going to get limestone in these shells? So I worked with as many museums as I have connections with to try to identify some known age specimens of these taxa, and I had pretty rigorous sample selection criteria. It was really important that we knew that they were alive when they died. So I started with a big long list of potential specimens. I worked with the curators, and we decided to just focus on the ones that actually had soft tissues in the collection as well, because then we knew that they were live collected. So that really narrowed down our sample universe to practically nothing. But we were also focusing on the post-bomb period, so that it would be more kind of sensitive to changes. And then we also were looking for specimens that had precisely known collection coordinates, so we could tell, looking back, were they from a limestone or non-limestone environment. And then also, at the request of the museum, I only focused on sample lots that had more than one individual. I didn't want to destroy the only one specimen they had from a given locality. So starting with a list of 100 lots, I ended up with a list of 11 really good candidate specimens. So I don't have a lot of data to show you, but it was really intentional sampling strategy. And here's a nice little map showing where they came from, mostly the kind of western side of Florida. And the hurricane's supposed to hit here-ish. Here's a very, very simplified diagram of the surface geology of Florida. I just color-coded it limestone versus everything else so that you could see how much limestone there really is in the environment here. Because our sampling strategy in terms of the location was just a little bit opportunistic, we don't have a really even sampling of limestone versus non-limestone. Most of them are from limestone environment, but we do have 2 that are from non-limestone, so that's really helpful. So I want to emphasize that we didn't date the soft tissues, we only dated the shell carbonate, and I took a little clip of the shell from right at the aperture at the opening, so it represents kind of right before death. The pink dots are the rosy wolf snails, and the green dots are the flat coils. And from a mile away, you can see that they do not plot on our bomb curve. For the most part, they tend to be below the line, which is suggesting that they are incorporating some very old carbon. But there are some other patterns. I know it's a small sample size, but there are some patterns you can kind of suss out here. The pink dots tend to plot closer to the line, and the green dots tend to plot a lot lower, and there's also a lot more variability. So we can estimate the percent of the dead carbon that's been incorporated into their shells quite simply. This is just a very simple model here. I'll keep this color coding where the pink is the rosy wolf snail and the green is the flat coil. The rosy wolf, typically less than 5% of their shell carbonate comes from the limestone or the dead carbon source, whereas the flat coil, it's really all over the place. And this does make sense in terms of their behavior and their biology. The rosy wolf tends to spend a lot more time in the trees. It tends to be more arboreal, and it will come down to the ground to feed, but it's not spending its entire life on the ground. So it's not as exposed to limestone on the ground surface. Whereas the flat coil is 100%, it lives on the ground, usually under, rocks and under logs and things. So it's not surprising that it would have more exposure to this limestone source. But the spread, the range of variability, I'm not quite certain how to explain that. I don't know if it's the amount of limestone at the surface or some other explanation. But this is pretty discouraging for archaeologists who are interested in dating these critters. So for rosy wolf, 5% dead carbon, that's maybe like a 400-year age offset, whereas 25% is really quite substantial. So here are the data. Looking at our map here, you can see that most of our critters are from the limestone environment, but there are 2 that are not, and these are the exceptions that prove the rule. These are the 2 specimens from the entire sample collection that had 0% dead carbon in their shells. So that is, to me, pretty compelling. I would like to have had more from the non-carbonate environment, the non-limestone environment, but this is what it is. So to me, it suggests that from a non-limestone environment, it may be possible to date these critters, but there is a big caveat to that as well. So when we're talking about dating these organisms from shell-midden sites, shell-middens are carbonate landscapes. Every single shell-midden is an absolutely unique biome, isotopically unique biome. So in terms of the fossil source of carbon, we can say this is dead carbon, this is where it's coming from, but when they're potentially incorporating carbonate from an older shell-midden site, that's really hard to account for. Even if they're incorporating carbon from shells that are of contemporaneous age, marine shells of contemporaneous age, they'll be incorporating some amount of marine carbon. So these are variables that are really difficult to account for. So my takeaway for archaeologists is buyer beware. These flat coil snails I think are going to be really problematic, and I would avoid those completely. The rosy wolf snails may be a little bit less of a problem. The limestone case would really represent the worst case scenario. So if they're interested in a range of variability of maybe, plus or minus a few hundred years, maybe these rosy wolves will be useful to them. But in general, if we're trying to get towards more precise chronologies, this is just adding layers of uncertainty to already uncertain situations. So this is kind of a cop-out, but the summary is that dating shells is really, really complicated. You've got to consider the specific local environment, the habits and habitats of these different species. It's really difficult to generalize or to borrow a concept from one area and apply it to another. You really have to do your homework and put a lot of work into just determining what are the sources of uncertainty, what's the range of variability, and what are the expected outcomes. So I often will see publications or reports in Southeastern archaeology that make some pretty big claims based on chronologies, based on shells. So I caution you to take some of these interpretations with a very large grain of salt. And at the same time, I will do my job to try to help educate archaeologists on the potential problems and pitfalls with radiocarbon dating these shell sites. I'm not going to discourage archaeologists from dating shell entirely because it is a very expedient, easy way to approach these shell-midden sites, and when they're being lost and threatened by hurricanes and things, something is better than nothing. But still, we have to be aware and appreciate what kinds of problems we might be seeing in our data set. With that, I thank you very much for your time, and I welcome any questions. Oh, I'm supposed to sing, right?
Yeah.
Right. Volare, oh, danzare, oh.
Okay, it's time for a couple of questions. Carla? Christine, sorry.
Thank you, Carla. I have a question about the calcium. If they don't eat the limestone, where do they find the calcium they need for the shell?
That's a really good question. The terrestrial ones that aren't incorporating? I don't know the answer to that. Does anybody have a suggestion? I do not know the answer to that, and I would welcome any suggestions that others in the audience might have. I don't know.
Okay, another question. Time for another question. Yeah?
Yeah, we saw the same effect on different species, but as strong as you have measured. I was wondering, did you see any signal in the delta C13? And because we were thinking maybe we can find a way to use the stable isotope to correct for this effect, but I never succeeded. And so I don't know if you saw something.
So we have the stable isotope data, and I can't quite get my head around it. It ranges from almost 0 to minus 25, and it doesn't seem to correlate with any of these age effects. So that's something I need to dive into a little bit more deeply. But there's C3 plants here, there's C4 plants. It could be getting it from the water. It could be getting it from the shell carbonate that's from the shell midden. So there's so many different sources. It would be hard to really constrain it. There's not just 2 end members.
Okay. Yeah.
As you mentioned, dating of shell is very complicated. I completely agree with you. But my question is, what kind of the component do you date? That's the calcite or aragonite?
It depends on what shell we're studying. So there are some that are biogenic calcite in this region, and some portion of the shell is calcite, but most of them, of course, are aragonite. We don't put a lot of effort, and I haven't put a lot of effort into really trying to separate the calcite from the aragonite in a shell that might have both. But for these are all modern shells, and I can't remember off the top of my head whether they're aragonite or calcite.
But I think the reliability of shell dating is should use aragonite rather than the calcite. Thank you.
I agree with that, yeah.
Any more questions? Okay, I had a question.
Yeah.
The terrestrial, the one is the connecting to the tree. It's something of the material from the tree as well, right? Or just the litter in the ground as well?
It just eats other snails.
Okay.
Yeah.
Yeah. That means they eat the carbonate and a little bit of...
Yeah.
Okay, thank you. Okay. Okay. The next presentation is by Rainer. He will talk about the radiocarbon signature of the Swiss River under the hydrological extreme.
Test,
Thanks.
test. Wo. Okay. So first and foremost, I would like to express my gratitude towards every individual one of you who has contributed to the advancement in radiocarbon measurements techniques, because this is exactly what allowed me to apply such a powerful tool within the natural environment, and this allowed me to really excel during my PhD with such a high number of measurements. So thank you, Arno. Within the context of global carbon cycle, I have highlighted the center of attention for today in red. This is within the terrestrial biosphere. Please note the significant flux and its associated uncertainties. To refine these uncertainties, rivers are very helpful. This is because rivers are natural integrators of watershed processes. They represent the entire catchment. You can imagine different carbon sources of old and young carbon being mixed downstream, so radiocarbon is a proportional average age of these different source end members. To understand how the terrestrial biosphere responds to climate change, sustained long-term observation of rivers is key. Luckily, in Switzerland, we have such a national river surveillance program where the government monitors 21 river stations since more than 50 years, but they don't measure radiocarbon. Here is where I come into play. So the Swiss Alps are changing rapidly due to alpine warming. If we focus on the 4 major river systems in Switzerland which drain the Swiss Alps, we can see a significant increase in river water temperature along with DIC concentration over the past 5 decades. The latter one, the DIC concentration increase, is very important because the DIC pool in rivers is an order of magnitude greater than both of its counterparts, POC and DOC combined. And here, thanks to radiocarbon and stable isotopes, we can show that at least 1/3 of this DIC comes from soil respiration. Thus, we are able to show that alpine greening leads to a stimulation of soil respiration rates, thus mobilizing DIC into rivers. And I am particularly interested in those underlying processes and what is really controlling them. Again, the map of Switzerland, station number 10, indicated as the purple star, is part of a 10-year monthly radiocarbon time series due to its proximity to ETH and Zurich. Here you can see the radiocarbon values. So for the organic carbon pool, POC and DOC, left and middle, you can see high variations. This correlates with seasonality, growing of vegetation, as well as the wet season, where rain events flush the fresh litter of the vegetation into rivers and so contribute to these modern radiocarbon signatures, whereas when you have an absence of rain events, you have a more pronounced headwater signatures with more depleted delta 14C signatures, which you can see with these spikes. But for Di14C, as you can see, it's very invariant. We think this is of constant groundwater input, of a constant carbon end-member ratio, or atmospheric exchange processes. Because I am measuring the same location every month, the water has the same time to equilibrate with the atmosphere. So I'm really interested if we see these observations also under extreme climate. This is just a recent study from Tibet showing the exact same story. As I told you now, in the middle upper graph, we can see how DIC is escalating, as they say, with decadal warming. On the left-hand side, we see the dry scenario with the bigger arrow in the groundwater input delivering DIC. On the left-hand side, we have the wet scenario where the arrows are bigger for the surface runoff, delivering organic carbon into the river. Unfortunately, they didn't measure radiocarbon. We have, and again, this is very crucial to link this decadal observation to a large-scale terrestrial biosphere. So during summer 2021 and 22, I have sampled all these 21 river stations. Here are some photographs of Swiss rivers. We usually sampled from the middle of the bridge. The upper middle photograph is from this 10-year-long time series. This photograph was taken under normal conditions, whereas if you have a decadal event of floods, it looks like this. So in terms of these extremes, I was very lucky. Here we have the hydrograph of 2021 and 2022. The different shades in colors represent different stations. And 2021 was characterized by a 30-year decadal flood event. And actually the subsequent following year, 2022, was characterized by a 30-year decadal drought event. So I was very lucky to be able to sample the exact same location under such contrasting hydrological conditions of climatic extremes. On the left-hand side, we have PO14C values plotted 2021 against 2022. And we can see along the one-to-one line a beautiful shift towards more modern values during the flood event. So really highlighting our observations, what I was talking before. Interestingly, for more than half of the DI14C values, the value is exactly the same, despite such contrasting hydrological conditions of climatic extremes. These observations are again supported by the strongest correlation between PO14C and water isotopes, really reflecting rain events, whereas for DI14C the strongest correlation was with mean catchment slope. Obviously, the slope doesn't change from one summer to another, but it really controls the water residence time, thus atmospheric exchange processes, but also groundwater input. Yes, only a few papers, in my opinion, really address this CO2 outgassing from groundwater. You have a substantial higher PCO2 in groundwaters. This leads to CO2 outgassing, which drives depleted delta 14C values towards more modern values. But also the same is true for CO2 uptake from the atmosphere. Here, DI14C values are more controlled by time and distance, also driving these signatures towards more enriched values. And yes, this is exactly my take-home message about non-conservative behavior of riverine DIC, where we have CO2 degas uptake, but also isotopic equilibration processes, and therefore, with regard to DIC, radiocarbon is not a proportional representation. In conclusion, this 1.6% increase of DIC per decade is really just a minimum, since DI14C values have this strong imprint of atmospheric CO2. Catchment characteristics such as slopes were a strong driver, while hydrological processes of these extreme flood events and droughts played more a subordinate role. Atmospheric exchange obscures isotopic signatures, really questioning and challenging the conventional approach of simple mass balance. Regional climate and topographic characteristics were dominant factors controlling water residence times, hence the downstream export of DIC. With that being said, vincerò, vincerò.
Okay, it's time for several questions. Okay, I have questions.
Yes, please.
When you have to say the groundwater, degassing, is it what you mean? Is it a change between the river water and the groundwater, or different?
It's exactly at the groundwater seepage, the interface between soil and the river. There you have the highest PCO2 pressure, and therefore it leads to a lot of CO2 outgassing.
To the atmosphere?
To the atmosphere, yes.
Okay.
And this signal is somehow lost, and it's difficult to back calculate how much was lost and what the radiocarbon signature of this loss was. Yeah.
Okay. Any questions? It's very quiet, right? Okay.
First of all, congratulations for the nice work, a lot of data you gave.
Thank you.
It is a simple, practical question. How do you do the sampling every month from the river? So it is a point sample on a day, or you have kind of integrating thing?
No, the radiocarbon measurements and the isotope measurements are all point snapshot samples, where we sample with a rope and a bucket from the middle of the stream. But the DIC concentration measurements, for example, from the government, they do a 2-week flow proportional measurement. And I checked if the 14C are similar, and sometimes yes, sometimes no, but we stick with the point sampling.
Okay, thank you.
So
Thank you for this interesting talk. In the beginning, you talked about rivers being integrators of the entire catchment, and at the end, you stressed that this may not be true for DIC radiocarbon. How about the other carbon components? What's your take on that? POC, DOC, how well do they integrate, and what would be the factors that may change this integration towards a point source or a localized signal?
I would argue a POC, since they're particulate, this really showed a strong correlation with surface runoff, discharge, as well as specific types of land cover, whereas DOC is somewhere in between. Since it's in the dissolved phase, it also travels through groundwater, and as you know, DOC is very biologically reactive. So I would argue that a POC is conservative, but DOC is somewhere in between, where you also have transformation processes of DIC into the DOC pool or the other way around. But the most non-conservative behavior is really seen in the DIC pool due to its gas exchange processes.
Okay. Okay. Thank you.
Thank you.
Okay, the next presenter is Zazorskaya. Sorry if I... Oh, she's not here. Oh, okay. Then we move to the last talk of this section. That is the presenter is Scott Cummings, talking about dead carbon in living individuals. Thank you.
Thank you very much for the opportunity to be here and talk with you about my research. We'll get to the reasons for some of the living tissue studies towards the end. The beginning of my studies with the presence of ancient carbon in living tissues began when I was asked to do some work in the state of Minnesota, which I'm going to guess. Nope. Okay. Which is here in the upper Midwest of the United States. And this all began because dates from charred food crust obtained from ceramics were coming back too old. In the United States, in this portion of the United States, there are no ceramics until the last 2,000 years. In fact, it's probably more recent than that. Thank you. So the area of ceramics, and is that? Oh, use this. Is here towards the center of the state. This is an example of all of the dates all of the legacy dates that were known by the time I began some of this study. It was expected that the bone dates would be most appropriate. The charcoal dates could be a little old because a lot of them are either on conifer trees or oak trees. And the charred food crust, so the charcoal dates, as you can see, all but one of them are more recent than 2,000 years on the charred food crust. And the charred food crust is the foods that have been burned on, and in this case, it's the upper part of the sherd, and this was decorated ware. So as the carbohydrates cooked, they would climb up and get burned in the drier and hotter area of the rim of the vessel. those dates tended to be at least 1,000 years old and as much as about 3,500 years too old. So many of those dates were simply recognized as this is not possible. We don't know why they're coming back too old. The bone dates intrigued me because of the 11 bone dates from sites containing ceramics, 8 of them were too old, either suggesting multiple uses through time of that landscape or a problem with bone collagen. Oops. I forgot that I timed that. Okay. Another site provided an opportunity to take a look at bones because this is a rock shelter in the southeast part of Minnesota. We had from this site only one piece of walnut shell, so one annual here, that came back very recent. Most of this blue area comprises either charred bone or calcined bone. The bone collagen may be observed in this area, or most of these dates that are older are in fact bone collagen. Only one was on calcined bone. So again, we're getting a pattern. The calcined bone should have been reset to the period of burning. So I expected it would have more uniformly recent dates than would the bone collagen if there was a problem in the diet of these animals. So the most logical place to look is let's examine the freshwater reservoir effect. So we did. And the geology, well, maybe I'll get to that later, but the geology in this state is divided. In some areas, the DIC signature is quite strong, and it's showing up in the fish. So the first 4 fish were, and I talked about this at the meeting in Trondheim, so some of you will have seen it. These varied from about 300 years, 307 to over 1,000. 1,223 years BP. These dates vary along the known trophic level of these fish. So the walleyes are known to jump. They eat a lot of flies. And as any fisherman or fisherwoman will tell you, that is how you catch them when you go fishing. So the carp, the sturgeon, these all are going down in the stratigraphic layers and the catfish feeding from the bottom. Seemed like, okay, maybe we're getting somewhere as to where the ancient carbon is coming from that is finding its way into the pots, because if you're cooking the fish, you're introducing these things, these ancient carbons. So those were all caught in 1939, were pre-bomb. They were curated at the state offices. So in 2015, I had asked, could I have some modern fish? Let's start checking out. So we got 2 northern pike. They are very carnivorous fish, but apparently they go up and down in the water column. They must have been feeding on something really old, because in spite of the fact, if you're going to catch them, you'll fish in the upper part of the water column. They have to be eating well below that. So then we looked at, okay, is the flesh of the fish, does that date about the same as bone collagen? So we looked at a catfish caught in one of the lakes, and the flesh and the bone collagen dated the same. I think it was very much the same, 530 years. And we looked at the stomach contents, which were older by 100 years. So just what you would expect. So things started seeming like they're coming together. I thought, okay, if you're going to look at charred food crust, what you hope for is the carbohydrates are boiling up to the rim, much like if you cook pasta, you get all of these bubbles at top, and if you're not washing your pot every day, you could burn those and have that as your charred food crust. So we looked at wild rice, which looks like this when it grows. It grows semi-submerged. And the part then that sticks out of the water, these are harvested by canoe. That's where you get the seeds. So we looked at that. The fraction moderns came back fairly old. These are from, I think, 2017 or 2018. So this lake and this river yielded more offset and more DIC than this location. So we're starting to get a handle on where in the state you would expect to have some problems. Then I compared the fraction moderns from even more fish bones that we examined and tried to show graphically how these might relate to the offset in the fish bones collected in 1939, the pre-bomb. This is just a very, it's just meant to be a graphical representation to make it easier to understand. So we continued looking at all of the things that go into creating the offset in fish. And so we looked at the humate from lake sediment, from the same lake, an aquatic broadleaf plant growing submerged. So the only thing that was up into the atmosphere was the top of the leaf. And then an aquatic grass. Aquatic grass sticks up higher, and you have more surface area for interaction with the atmosphere. I turned to better understand the bone collagen and gelatinized collagen to a study done by Tom Stafford in the beginning in the 1980s and then continuing into the 2000s. And when he used the XAD method, he got his oldest dates here, but he also then took the purified bone collagen, gelatinized, and separated into the various amino acids. So I became intrigued by the variability noted in those amino acid fractions. And as I started to look into the amino acid fractions in collagen and just verifying, yes, it's mostly 50% are glycine, proline, and hydroxyproline. And I started to wonder, because these are all non-essential amino acids, I wanted to start looking into how does the body build these amino acids? Where is it taking the carbon? So we'll use the example, if I'm eating a salad or rice or pasta or potatoes or meat or fish, I'm getting carbon with every bite. How does my body separate these things, or does it? And does it use, I mean, it's just, where do all of these things combine in the body, or are they held separately? So I started looking into proteomics, and I found someone who was quite interested in helping me delve into this until he told me, oh, but I don't understand metabolism. Now you need to talk to my colleague in metabolomics. And I thought, oh my gosh, now there's too many omics for me to deal with. So the course of talking and finding the right people means when I return, I will pick that conversation back up again. I haven't had it yet. I've not met the metabolomics person, but we'll get there. I'm sure we'll get there, and this will be an ongoing study. So, how did I get this far? Well, I thought maybe I was at kind of an impasse or needing to go back to some of the fish to study from Minnesota. Well, then it happens that I found out I needed hip replacement surgery. And I thought, ooh, this is an opportunity. So I asked the surgeon. I said, may I please have the part of the head of the femur, this part you're going to cut off and throw away. Can I have this? And he said, do you have a dog? I said, no, I don't have a dog. Oh, good, you're not asking for this so that you can give this to your dog. I said, certainly not. So I told him that I wanted to radiocarbon date my bone because I know my history and I know what I eat. And I thought I had a varied diet. I eat fish and shellfish and all sorts of things. So I thought I was going to get an offset, much like Peter Grutas reported at the Trondheim meeting. No such luck. I don't know. Maybe Colorado does not get good seafood. Maybe I don't know what it is. So so these are my results reported. So, and I chose to do this because it was too cluttered on the bomb curve. It was too hard to see what was going on because everything is too similar. So, turnover rates look like these dates, if I had a typical center of the country diet, look like they would be about appropriate. Turnover dates of about 20 years of my collagen, and on my appetite, faster turnover, more like 15 years. The muscle came in, so I organized these by chronological date rather than by the part of me they represent. Oh, and by the way, I decided not to show the pictures of the bone because I felt when I asked a few people, the results were, oh yes, I want to see that too. Oh, please don't do that. Please don't, no, I don't want to see that. So if you want to see, get me later, they're on my phone. So I went through the typical defatting and all of this. So we collected the lipids, and that had the fastest turnover. The surgeon had left a tiny bit of muscle. I took that off, and we dated that as just something that was not bone, just out of curiosity. So I thought, oh, darn, I can't use my own bone to go forward to do this examination of what do the individual amino acids look like. So it's back to the curated collection, request the fish bones yet again from 1939, and we'll start with those to see, is there anything that we can learn from examining the individual amino acids, and do they all cluster at the same level as the collagen data, or are we going to see variability? Because if there's something that comes back more towards the expected age of the fish, maybe that's something that we can extract and do a better job at getting congruent dates from the archaeological record. Thank you for your attention.
Okay, it's time for several questions. Yes, over there.
sit over there, but it's a very short one. When was your surgery? What year?
This year. So,
This year.
left hip in February of this year, right hip in June of this year. So,
And the fingernails, when did you cut them? This
what-
year?
This year, when yes. Unfortunately, I had come back to work and I didn't cut my fingernail first, and by the time I cut it, I had already been working with some charcoal samples. So I didn't focus on the fingernail because I think maybe there's a problem with it. Who knows?
Because you are on the other side of the bone peak now.
I know.
With your
It was
fingernails.
the other side. And the only explanation I can come up with is, okay, when I work with charcoal, you do this. You snap and you use your thumbnails to help snap. That was the biggest nail. I mean, I should really have cut that nail before I returned to work.
The question over there, and then Tim is over there first. Yeah, then Tim. No. They have a microphone there. Yeah, please.
So in terms of publishing data on your own skeletal remains, is there anything you have to think about in terms of human subjects research and getting any kind of like IRB approval? Like, how does that work?
I asked the surgeon if there was a problem with me including him as a co-author, and he said no. And in fact, when I asked for permission, he said he would have to ask the hospital. He came out of surgery holding a brown paper bag, and he gave it to my daughter and said, "Here, this is for your mom." So he decided that my reason was good enough. He has told me, no, you'll have no problems.
Okay,
We'll hold.
Tim. Yeah, a question for Tim. Yeah.
It's a comment. So the Grotus paper is, yes, there's a question about IRB approval. So if you publish it, you might want to think about that. The second thing you might want to think about is that because you gave us the amount of C14 in the bone and in the tissue, you can probably calculate your age. So just
Go for it.
Okay, any more questions? Okay, I have questions.
Yes.
Okay, then you have shown all the results, and what advice when you have the archaeological sample with the fish or in the river, then what you advise? What you do?
Oh, yes, I'm working through that. So I think at this point, I advise the state of Minnesota that we need to look more into the DIC and radiocarbon date the individual watersheds, because there's 5 or 6 for that area. There's some parts of the state that seem good, and other parts of the state have too much DIC, and we're getting too much reservoir effect. But so what I have told the archaeologists so far in the state is you're probably going, depends on which part of the state, you could easily get dates that are too old on bone collagen. Prioritize the charcoal, date that instead. And then go with calcined bone. So we're still trying to narrow down which parts are where you can rely on the radiocarbon dates and where you will end up thinking that these cultures are very, very ancient. And so it's still a work in progress.
Okay.
And I'm thinking that over the next 5 years, those are the questions I want to answer.
Okay, thank you. Is there any more questions? Yep. Elizabeth?
You were talking about essential and non-essential amino acid.
Yes.
because there are some amino acids that we get straight from food and we don't have to. I think Robert Edges published a paper that he tried to use the essential amino acid that we get straight from fish and see if there was a way to correct for reservoir age due to diet. Have you seen this work? It was on the river, I think the Donau River or something.
Yes, it's one of the papers that I use as in an argument for why it's now necessary to begin to look at the essential and non-essential, but to find enough of the essential amino acids in anything archaeological could be problematic because each individual amino acid, if all of the rest of the non-essential and essential amino acids comprise only 50% of the collagen, you could probably, and there are none of them that are even 12 to 15%, so they're all going to be really a low percent. So yes, I want to look at it in reference. I don't know if it will have, in fact, any validity for archaeological materials because of preservation, but I hope.
Okay, if we don't have any question, I would like to close the session. And could you please to join me to thank all the speakers in this session? Okay, it's lunch time now. Okay, you can enjoy lunch.
Okay, now the time for the session is approaching, so please have your seats and we'll listen to some amazing talks in the session of atmospheric and terrestrial environment and the interactions in between. So let me invite the first speaker, who is Andrew Smith from ANSTO. He's going to tell about the microsamples for micro C14 measurements in methane.
People are still having lunch. I'll make a start, nonetheless. And I realised when I was preparing this talk that I had set myself an impossible task. The task of trying to summarise about 25 years of research into a 20-minute talk. So I apologise in advance. I'm almost certainly going to run into question time. However, there'll be plenty of opportunity over a glass of wine to ask any questions if they'll keep that long this afternoon. Could you please give me a 15-minute warning when we get there? Okay. So without any further ado, if I can work the system. Before I talk about the applications, and I want to talk mostly about the applications, I'm going to give a little brief overview of what we call microcarbon AMS in the Centre of Accelerator Science, which is part of ANSTO, Australian Nuclear Science and Technology Organisation. And at ANSTO we use solid ion sources. So we convert the carbon dioxide into graphite using the Bosch process, so that's 600 degrees, an ion catalyst in an excess of hydrogen, and we operate banks of these reactors to produce the bulk of our samples. I'm going to highlight a couple of rather specialised reactors we've produced just to produce the very small samples that are involved in this kind of climate research. Our targets are rear-loaded, as I think most people's graphite targets are, to a reproducible depth and a reproducible surface finish, which is absolutely critical for measuring tiny samples. And we use the largest of our particle accelerators. We've got 4 accelerators at ANSTO, the 10 million volt Antares, 6 million volt Sirius, 2 million volt Star, and 1 million volt Vega accelerators. But ironically, we use the largest of the accelerators for these micro samples. And the main reason for that is that accelerator is equipped with an 846 ion source, HV 846 ion source. And this ion source is both a blessing and a curse. It's a curse in that it is prone to destroying itself, but it's a blessing because it allows you to center the beam, the cesium beam, right in the middle of the target. And once it's centered, the target can then be rotated under the cesium beam. And what this means is that basically all of the carbon in the sample is consumed during the sputtering process. So we make maximum use of the available sample size. And you can see down on the bottom right there a procedure that we use to do this centering. It involves using our copper and aluminium targets and doing a mass scan. There's the accelerator on the left, the big FN 10 million volt machine, and the 846 ion source. We've just recently relocated these micro furnaces to a new laboratory, and you can see there the layout of the lab. I should point out the most important thing in this photograph is actually in the middle, and that's Bin Yang. Without Bin and his abilities, we would have struggled to have undertaken a lot of this research. So over on the left there is the prototype LHF. So LHF is a laser heated furnace. So I developed that some years ago. That's the prototype device. It's a rather big box. And inside that box is the reaction chamber, and the ion catalyst is heated by a focused infrared laser. So the infrared laser is focused to about half a millimetre diameter spot, and it's focused onto the ion catalyst, which is contained in a small quartz crucible in a very tiny volume. So the whole design of this furnace, it's designed to minimise the volume because, of course, the Bosch reaction is pressure dependent. When you've got small samples, you've got small pressures if you've got big volumes. So we wanted to keep the pressures high, and that speeds up the reaction process. And this furnace also has an integral welded cold finger. So it's a rather different design to a lot of the furnaces that we've seen in this talk so far. And it can do samples from 0 up to 100 micrograms of carbon. And the whole graphitization reaction, I might add, takes place in about 20 to 30 minutes. It's very quick. And because only basically the catalyst is heated, there's a very small heated surface inside this furnace, and it means that the extraneous carbon, that is the carbon that we don't want in our sample, that's introduced during the graphitization process, is minimised. In fact, it's very small, as you'll see. And Bin has taken the basic concept of the LHF further. So you can see if I can get this thing to work. So it's a bit shaky. So these two furnaces here are the second generation laser heated furnaces. And over here, there's a bank of 6 what we call micro conventional furnaces, MCFs. And these are have changeable cold fingers. So that means they can also deal with very small samples because the reaction volume is small. It's less than a milliliter with suitable configuration of cold fingers. But it can also deal with samples up to 2 milligrams of carbon. And in fact, those are really our workhorses. And most of the climate change stuff I'm reporting on in this talk is actually, they're actually graphitized in those furnaces rather than the LHF. But I'm going to talk still a little further about the LHF. So that histogram there summarises sample sizes for the last couple of years for our cosmogenic in situ dating program. So Reka Fulop, some of you may know, extracts carbon from quartz grains for in situ dating and has a very rigorous cleaning procedure to get rid of atmospheric carbon-14. And although it's probably impossible to read that scale down there, each of those bins is a microgram. So the smallest bin right over on the far side of the screen, that's a microgram. And you can see, I've got to put my glasses on. You can see that 15 samples in this over the last couple of years have been less than 1 microgram of carbon, and the bulk of them have actually been less than about 5 micrograms of carbon. There's an illustration of the monolithic construction of the furnace. So key to making it small is actually machining it out of a block of steel. It doesn't look like steel, but it is, and having integral valves. And as I say, this stainless steel capillary, which is laser welded into the system. So basically, the top of the reaction chamber is sealed with a quartz window. The quartz window is transparent to the 808 nanometer focused infrared laser that heats the catalyst, and it's also transparent to the black body infrared radiation that catalyst produces as it's heated, and that's monitored by an infrared thermometer, which views at an angle through the quartz window. Anyway, that's probably enough of that. Here's the performance of the LHF. This is again over the last couple of years. So at ANSTO, we produce a lot of reference standards. We typically normalise to oxalic 2 because it's more active. You get more counts, more bang for your bucks. And we also regularly produce OX1, C7 and C8 reference samples, and we make them, they're commensurately sized samples, which allows us to do the data reduction. I can't go into any depth about that, but I'm happy to talk about it later on. And so a typical sample carousel, which holds about 60 targets, is often 2/3 samples and 1/3 reference samples and blanks. And you can see in this larger plot over here, I should also explain that these results are warts and all. So some of these are clearly outliers there, and they weren't included in the final data analysis. So you're seeing everything, nothing's hidden. Here you can see just a linear regression to 1 over the mass. So that's the normal sort of performance that we expect to see as you add extraneous carbon. And that's equivalent to the addition of, and that's over several years, 0.08 micrograms of 100 PMC carbon. So that's very small. In actual fact, both types of furnaces add of the order of 0.05 micrograms per graphitization. And that allows us to do this kind of work. Okay. Then again, a similar plot, similar set of plots this time. This is actually the results of reference samples from our hydroxyl network program that we've been running with the University of Rochester. And in this case, all the samples are 50 micrograms size. So let's get into the research projects before we run out of time. These are all externally driven projects, but they utilise our microcarbon capability. And you can see there the principal collaborators: the National Institute of Water and Atmosphere in New Zealand, CSIRO in Australia, University of Rochester, and the Scripps and Oregon State Universities in the States, and the University of Bern. But there are plenty of others as well. And the samples that we've been studying over these 25 years are derived from both the Arctic and the Antarctic ice sheets, and also from a global network of monitoring stations. So we're looking at both the present and the past atmospheres. Funding is from the National Science Foundation primarily, also the Australian Antarctic Science Program, and some specific programs from the Packard Foundation, and the most recent one from VESRI, which is part of SMIT Futures. That's the obligatory slide of logos. And this is a list of some 13 principal projects. It's not all of the projects that we've been involved in. And obviously, there's no way I can go into any of these in any detail. I would need an hour plus to speak on any single one of them because they're all quite complex. But I guess the important point is that this all started around about 1996, a little over 25 years ago. And I'm going to talk a little bit about that because it gives some illustration of the kinds of problems that are involved in doing studies in polar ice sheets. And also want to point out 2002, where we showed quite conclusively that radiocarbon monoxide is produced in sample cylinders. And that process is exactly the same one that produces radiocarbon in the atmosphere. It's neutron capture on nitrogen. The big difference, though, is in a gas cylinder, there's no ultraviolet light. So there's no hydroxyl radical. And what this means is that the concentration of radiocarbon monoxide can get quite high. And that's an issue because you've got to get rid of carbon monoxide when you're processing for methane. I'll explain that later. And I'll explain, and I'll highlight some of these studies as we go along and give you an idea what's been involved. And I guess importantly, this one at the bottom, Fetch 4, this is the fate emission and transport of methane. That's currently running. In fact, I was measuring samples for this program last week from, again, from a global network of atmospheric monitoring stations. And that's going to run through till 2027 or so. It's continuing the sort of work that we've been doing up until now. So, the beginning of the story, 1996. I was lucky enough to be tapped on the shoulder by my colleague Dave Etheridge and say, "Do you want to come to Antarctica?" And I said, "Yeah, that'll be good." And so off we went. And the goal of this expedition was to recover air from the Föhn. So the Föhn, the air from the Föhn for doing radiomethane studies to try and learn more about the sources of methane. And the Föhn is the compacting snow over the impervious ice layer. And the process of actually trapping air as bubbles in ice is quite complex because the Föhn is effectively a diffusion column. And you can see here that the depth at which the Föhn turns into ice is somewhere between 60 and 110 metres beneath the surface. It depends upon the site, the conditions of the site. But until you reach that point, the little interstitial spaces between the compacting snowflakes are still in contact with the atmosphere, albeit through paths of increasing tortuosity. I love saying that. But that means that what that ultimately means is when the air is entrapped in bubbles, the air in those bubbles is always younger than the ice that contains it. And furthermore, each atmospheric gas, because it has a different coefficient of diffusion, has got a different age offset. And also, there's not a single age, there's a range of ages. And all of this has to be modelled and deconvoluted before you can make much sense of the record in the ice bubbles. And my colleague Kathy Trudinger and Christo Buzet at Oregon State University both run Föhn air diffusion and trapping models, and part of running those models is tuning them. And back in 1996, we had the idea of using the radiocarbon dioxide bomb pulse because it's a rapid onset signal with a very known fall-off. And when you reconstitute that signal, as read in the ice core record, it's a very powerful constraint on some of the adjustable parameters in these models. I should also explain too that it's often necessary to take into account the fact that ice isn't static, it's moving, it's flowing. And so as you drill down into the ice core, you might not, or you will not be sampling ice that fell at that site. It'll be uphill somewhere, up the glacier. And so you need to have these kind of glaciological models to actually deconvolute the signal. Here's a little schematic of what's involved in doing fern air sampling. Basically, you drill a hole down into the ice sheet to a certain depth. You take your drill out. You insert a fern air sampling device, which is a big inflatable rubber plug. You block off the hole. You pump up the air, and you put it into sample cylinders at the surface. And then you continue the process. And you can actually get hundreds of litres of air in this way. It's not difficult. It tends to come in from a horizontal stratum. So fern air sampling is easy. But as you get down into the ice, you need to get the air out of the bubbles. And this picture here shows the ice grating flask used at CSIRO. And basically you load that up with about a few kilos of ice. You evacuate it, and it's on a paint shaking table, and it shakes the ice against the grater, breaks it into millimetre-sized pieces, liberates the air, and it's collected in an apparatus like this. But you're very limited, of course, in the amount of air that you can get that way. And typically you get something like 100 millilitres of air per kilo of ice. Oops, sorry, something went wrong then. No, okay. Then the story gets a little bit more complicated, because you're not just looking at C14 that came from the atmosphere. You're also looking at C14 that's been produced in the ice itself by the spallation of the oxygen. And that's caused by both neutrons and muons, fast muons and negative muons. Now, the neutrons don't penetrate very far. Typically, neutron production is over after about 10 metres of water equivalent depth, which is about 20 metres of fern, whereas the muons penetrate a lot further. And so in order to do these kinds of studies, especially when you're talking about methane, which is present in very low concentrations in the air, you've got to understand about the production of in situ 14C and what happens to it. And it's been known for a long time that the hot 14C atoms that are produced by these reactions, excuse me, so I'll just pause for a drink. The hot radiocarbon is rapidly oxidised into carbon-14 monoxide and then into carbon dioxide, but a small fraction is also converted into methane and indeed into other organics. And understanding the fate, the production proportions and fate of this in situ carbon-14 has eluded a lot of researchers. I talked about it in that 2000 paper. And there are three main reasons for that. And that is that sites with different accumulation rates have got different shielding for the neutron production, with the muons still continuing on through. We also have shown that you get a lot of venting of this in situ C14 in the fern column before it gets trapped as bubbles in the ice. Yeah, okay. There are different gas extraction methods used: milling, grating, melting, and sublimation. So I've actually used up my time pretty effectively. This shows the production in blue of neutrons. You can see it falls off very quickly. This is with depth along the horizontal axis, and the much more penetrating muons that continue to produce C14. We know a lot about methane. We know why it's important. And this is, of course, why we've been studying it, and we've seen this slide many times before. The important message from this slide is that you can see there's a big disparity between the bottom-up and the top-down estimates of the various sources of methane, and indeed of the sinks. And the atmospheric levels are a balance between the emissions and the sinks. So we have a lot still to learn about methane, and hopefully we're contributing towards that. Natural processes of methane, human processes of methane, each one has a different isotopic signature, and we've heard already today that del C13 and delta D aren't that helpful necessarily at distinguishing between these different sources. Carbon-14, though, is very powerful because fossil sources are depleted in C14. So to understand the methane sinks, excuse me, most of the hydroxyl most of the methane is actually taken out of the atmosphere by the hydroxyl radical, about 90% or so. And so we've been we've been trying to reconstruct the hydroxyl concentrations both in the present atmosphere and the past atmosphere. It's no longer possible to do it in the present atmosphere with methane chloroform because the levels have dropped to the point due to the Montreal Protocol where it's immeasurable. But carbon-14 monoxide is produced in the atmosphere all the time at a fairly known rate, and it's also removed from the atmosphere by the OH radical. So measuring OH, you can infer the concentration of OH by measuring 14CO. But this is hard. So I've used up my time, right? Oh dear, dear, dear. This is not good. So look, I'll just flick through these slides so you can get some idea of what's involved. That was fern air sampling in 97, 98. And that took us back to about 1935 AD. But of course, if you want to go further back in time, you need to get air from the ice. And this becomes very challenging. You need to be able to extract the methane from the air. The first part of that process is getting rid of the carbon monoxide, because the carbon monoxide will be combusted along with the methane into carbon dioxide, and carbon monoxide in air can be 800 PMC. And so even a small leakage of carbon monoxide through a processing line such as this in the University of Rochester will cause issues. So how do you get old ice? We went to a site in Packetsock in Greenland to look at the reasons behind the big increase in methane from the older dry ice to boiling transition and from the younger dry ice to pre-boreal transition. And this is the site. It's on the margin of Greenland. And I just want to point out this diagram here. This is a sort of a cross-section of an ice sheet, and those lines there are flow directories. So when you're on the top of the ice sheet and you're drilling down, you've got to go deep to get ice. However, as that ice moves towards the margins of the ice sheet, all of a sudden it becomes horizontal. So when you're walking across the surface at Packetsock, you're effectively walking back in time. And that means that you can access very large quantities of ice at the surface. New ice, old ice, ice movement that direction. That little orange dot there is a tent. There it is again. You can see very clearly the difference between glacial and interglacial ice. The glacial ice is dirty because it's dusty. And this is the sampling that went ahead in this site. This was not an ideal site, as you can probably tell. That's Jeffrey Severinghouse cutting ice blocks with an electric chainsaw. And the work that we did at that site actually showed that those rises in methane at those terminations was actually not due to the destabilisation of methane clathrates, there was the clathrate gun hypothesis at the time. A lot of ancient methane are locked up around the margins of the coast under the water where it's cold and highly pressurised, but rather it was due to biogenic production. We repeated this process at Taylor Glacier, which is a much cleaner and more pristine site, and came up with the same findings. So look, I'm going to wrap up soon. Just two theses. This one, Benjamin Hamil. This work went ahead at the summit of Greenland. There were 3 sampling expeditions. I was lucky enough to be on one of them. And the main findings from that thesis were that pre-industrial methane showed that there was a much greater anthropogenic fossil fuel contribution today than bottom-up estimates would suggest. And also we were able to get a much better handle on the in situ 14C production in the ice sheet and just what happened to it. And that's allowed us to do a lot more studies since. Another thesis, Michael Dionysius, these are both at the University of Rochester, was again at Taylor Glacier. So this is a horizontal ice core site. So the ice has come back up and you walk along the surface. That was my tent, that's where I lived. And you can see there the transept the sampling transept across the Taylor Glacier, which is flowing down the valley. Oh, sorry, I should just say that there were 3 sampling expeditions there, and the main outcomes from that were that old carbon reservoirs were not important in the deglacial methane budget, and we got a very good handle on the in situ production of radiocarbon by muons. And we continue this work trying to understand the hydroxyl radical concentration in the present atmosphere using sampling stations, and also in the past atmosphere at Law Dome in this case. And that was a big expedition in 1819. That involved not 1, not 2, but 3 different kinds of ice drills. And I don't have the time to explain that, but I'm happy to describe it later. And currently, Fetch 4 is ongoing, and that's going to keep us busy for some time, and probably another expedition to Greenland to recover some ancient air. And just finally, a project that's going ahead, an expedition that's going ahead this November, which I may yet be on, I'm not sure that I really want to be on it, is actually using some of the ideas that we've developed over this time to turn the whole argument on its head, and to basically, we aim to reconstruct the high energy component, at least, of the cosmic ray flux over the last 7,000 years by using a 14CO production. To do that, we need to go to a rather different site to the Law Dome site. We need to go to Dome Concordia, about 1,000 kilometres inland, where the accumulation rates are very low, and where the temperatures in summer are minus 22 to minus 45, and we'd be living in tents again. So I better finish up before I get booted off the stage. And I just...
Okay, oh. Yes, please. Still have some seconds to sing if you wish.
Yeah, I need to sing, but I can't make him... Why can't I make him sing?
You have to do it by yourself.
I have to do it myself.
Yes.
Volare, oh.
Okay, thank you for this impressive presentation of for such a challenging sites, challenging samples and analysis. I think we may have some questions, but we will move it for some breaks or the evening, perhaps. Thank you again, and I would like to invite Ivan Kontul from Bratislava to tell about the carbonaceous aerosols and their C14 signal in some very tiny fractions, and the sources of these aerosols. Yes.
Thank you for the introduction. So in this presentation, I would like to show you the results of radiocarbon analysis of carbonaceous aerosols collected in Bratislava, Slovakia. So the motivation for this study was that it is a logical extension of already a long-running aerosol sampling and analysis program at our university, focused mainly on radioactivity measurements. And we did a pilot study in 2019 where we looked at the source apportionment of elemental carbon fraction of aerosols. And now we wanted to look at the total carbon fraction of urban aerosols and to determine the proportion of fossil and non-fossil sources and the temporal variation. So we did a sampling campaign in 2022 and 2023 to collect aerosol samples in Bratislava. This is a satellite photo of the city, and here you can see the location of the sampling site, which was the campus of Comenius University. We collected the aerosol samples on quartz microfiber filters, And because we didn't use any pre-filter or impactor stage, our samples represent the total suspended particles in the air. And we were aiming for a 2-week sampling period for each sample, but 2 of the samples were collected during 3-week periods, and there is one 1-week sample, but most of the samples were collected during 2 weeks, during which we pumped between 1,500 and 1,600 cubic meters of air through the filters. Stopped working. Okay. So the quarter of the exposed filter was used for radiocarbon analysis. So we combusted the aerosol samples with copper oxide and silver wool, and the resulting CO2 needed to be purified to ensure a successful graphitization, for which we use the hydrogen graphitization method and in a reactor, as you can see. And then we, of course, analyze the samples using AMS. When it comes to AMS measurements, most of the measurements were done on the AMS system at the Center for Applied Isotope Studies at the University of Georgia in USA. But we were able to measure several samples at our radiocarbon laboratory in Bratislava, because in 2022-2023 our 3 MV accelerator system was upgraded with a fast bouncing system and the whole high energy AMS beamline. So we were able to measure several of the aerosol samples in one of the first sample runs on our new AMS line. We used blank preheated filters with synthetic graphite for blanks. And here you can see the results of radiocarbon measurement. So we analyzed 16 samples, and the PMC values ranged from about 70 PMC to roughly 85 PMC. During the sample preparation, we could also determine aerosol and carbon concentration in the sampled air. So the aerosol concentration ranged from 4 to 15 micrograms per cubic meter of air, and the carbon concentration that ranged from 1 to 4 micrograms per cubic meter. And you can see that the pattern of the carbon concentration is virtually the same as the pattern of the aerosol concentration, which means that the carbon content of these aerosol samples was quite stable, and it ranged from 25 to 35%. For the source apportionment, we used a simple 2-source model. So we are considering 2 sources of carbon in the aerosol samples with a different C14 signal. So the first is the carbon derived from fossil fuel combustion, which is radiocarbon free. And then it's the carbon from non-fossil contemporary sources. And mixing of these 2 kinds of carbon creates the observed or measured PMC value. And using these 2 equations, we can then calculate the fraction of fossil carbon and fraction of contemporary or non-fossil carbon in the samples. For the PMC value of the non-fossil carbon, we use the value of 105 plus or minus 5 PMC, because we, for Slovakia and Bratislava in particular, we don't have any data or information about how much biomass is used for energy production or domestic heating, and what fraction of that is made up of fresh biomass and wood. And because we don't have this data, we chose this range from 100 to 110 PMC to represent the uncertainty in this parameter. And these are the results for the 16 samples we analyzed. So each bar represents one sample, the width of the bar represents the sampling period, and the gray area is the fossil fraction, and the green area is the non-fossil fraction. So you can see that when we look at the non-fossil fraction, it ranged from 0.67 to 0.81 with a mean value of 0.72, which means that on average during this year 72% of carbon in the aerosols was from non-fossil contemporary sources, which appears to be quite high for a city that is highly both urbanized and industrialized. But if we compare these results with similar studies from other European cities, I think especially a good comparison can be made with Debrecen in Hungary and Zurich in Switzerland, because they are cities of, I would say, comparable size with continental climate. So we can see that the range we observed in Bratislava is comparable and consistent with these two cities, because in Debrecen it ranged from 0.6 to 0.8, and in Zurich from 0.6 to 0.7. Comparison can also be made with Budapest, although it is a much larger city, but it is in a similar geographical area. And there the range starts at lower values, probably indicating a higher fossil load, but the higher end of the range is similar to what we see in Bratislava. It needs to be noted that this comparison can only be done qualitatively, because these studies focused on a more specific size fraction of aerosols compared to our total suspended particles. But what I think is interesting about these results is that if we look at the temporal variation, so there is some temporal variation, although in quite a narrow interval of values, but there is no obvious or statistically significant clear seasonal variation, because the range of the non-fossil fraction for spring and summer is from 0.67 to 0.75, and fall-winter ranges from 0.69 to 0.81. And also when we look at the mean values for spring, summer, fall, and winter, we can see that there are really no differences between the mean non-fossil fraction for the individual seasons. For example, when we compare it with the Hungarian studies, which were also year-long sampling campaigns, there was a clearer seasonal variation in those results compared to what we see here. We can also compare these results with the previous study we did on the elemental carbon fraction on samples from 2017 and 2018, where we saw a much higher contribution of fossil-derived carbon, as was expected, but also we saw much clearer seasonal variation in these fossil and the fraction of carbon coming from fossil fuel combustion and biomass burning. So if we assume that this is a representation of how the fractions change within a year, it would seem that the addition of the OC fraction evens out the seasonal variation, and but this is something, if we want to study in the future, we want to look at the or make a synchronous TC and OCEC analysis to really see in samples from 1 year how the proportion of EC and EC changes and how the radiocarbon content of these fractions changes during the year. So just to summarize, these were the results of source apportionment, result of source apportionment of total carbon fraction in urban aerosol from Bratislava. And we observed a high contribution of carbonaceous particles from non-fossil sources, which are the burning on biomass and biogenic emissions from biosphere. And we didn't observe any clear seasonal variation because there are only very small differences between spring, summer, and fall-winter periods. So for the next, because the sampling of the aerosols continued and still continues, so we want to, in addition to radiocarbon analysis, we are also doing particle-induced X-ray emission analysis of these samples to determine the elemental composition, and because we want to see if there are larger differences in these other elements compared to radiocarbon content. And also we would like to do OC-EC separation and radiocarbon analysis and focus on the different size fractions and to see if the radiocarbon content, how the radiocarbon content changes during that year for these different size fractions of aerosol particles. So that would be everything. Thank you for your attention.
Thank you. So, are there any questions? Elizabeth?
Sorry? I think it would be the biogenic emission would be even higher because even now it makes most of the carbonaceous particles in the atmosphere, and during COVID I think the fossil load was lower, so yeah, I think it would be even higher the contribution of the particles from non-fossil sources.
But you don't have samples.
No, not for this period.
Okay, thank you. Are there any other quick questions? Okay.
Isn't it a little surprising that the non-fossil fraction is that high in the urban areas?
Yeah, it was quite...
is that an explanation, or is this expected, or other urban areas have a much lower fraction of that?
Yeah,
do you know?
We didn't expect that, especially in the EC fraction that comes solely from, so it's the soot from incomplete combustion, so it comes only from fossil fuel combustion or biomass burning. And there we expected to a much higher fossil fuel derived fraction. But one aspect that can play into this is that I think one of the districts of Bratislava uses waste wood chips during heating season for heating, for energy production. So I think this contributes to the fact that it's a little higher. Also in the autumn, burning of agricultural waste can contribute to higher biomass burning fraction. But yes, it was unexpected. But for the TC fraction, it's comparable with studies from other comparable European cities. So,
Okay, thank you. I cannot see any more people, so let's thank Ivan. He
Thank you.
didn't sing, so sorry. Okay, so let me invite the next speaker, which is Zhenchuan. Is it correct? Okay, new. From Xi'an in China, and he's going to tell us about the radiocarbon in carbon dioxide, but in the Quinning Mountains. Okay. Oh, yeah.
Hello, everyone. My name is Zhenchuan Niu. I come from Institute of Earth Environment, Chinese Academy of Science. My institute located in Xi'an, the central of China. Welcome to Xi'an. Today I show you a part of my work. There are three parts in my presentation. First is the background and objectives. Atmospheric CO2 observation is very important. From this observation, we can understand the current status and changes in atmospheric CO2 concentrations. We can understand the carbon cycle process and understand the relationship between CO and global warming. We can also use this observation to invert carbon emission and carbon sink. There are more than 300 site observations to observe CO2 concentrations. However, most of these sites are located in the United States and Europe. And the observation sites in China are insufficient and mainly on ground CO2 concentration. And the vertical observation and the observation to quantify the source are still rare. The inefficient observation site in China will affect the accurate carbon sink emission in China. A paper published in Nature said the carbon sink in China is about 1.1 pg carbon in China. However, most of the research think this value is overestimated. So we need more atmospheric CO2 observation in China, especially in some important regions. Qinling is located in the center of China. It's an important geographic borderline of the North and South China. This region with abundant vegetation. From this study, we want to know what's the level and variation patterns of atmospheric CO2, which source influence the atmospheric CO2 variations, where does the atmospheric CO2 come from? The observation site is located at the north foot of the Qinling Mountains, the edge of Guangzhou Basin. There are two plans in this study. First is the temporal observation. we observed the atmospheric CO2 continuously on a tower using Picaro instrument from March 2021 to February 2023. We also collect air samples periodically to measure the radiocarbon. The second plan is the vertical observation. Airbag was collected after different times in one flight using the unmanned aerial vehicle. The height includes 20 meters, 100 meters, 200 meters, 400 meters, 1,000, 1,500 meters, and 2,000 meters. The sample were collected at 4 times a day. It is at 8 o'clock, 13 o'clock, 18 o'clock, 23 and half o'clock in a day. We measured the CO2 of each sample and the 30 carbon and 40 carbon in the lab. From the two observations, we can find that the CO2 concentrations at this site is increased. It's increased, and with a mean value of about 442 ppm, it increased about 4.7 ppm compared to the previous year. For the diurnal version, we can find that summer has the largest diurnal amplitude. The diurnal variation trend of CO2 concentrations on workdays and non-workdays were consistent in each season, but the diurnal CO2 concentrations were generally high on workdays than non-workdays, especially in winter. For the monthly variation, we found that the highest CO2 concentrations in January and the lowest in June. There is also obviously seasonal changes, highest in winter and the lowest in summer. From the vertical observation, we can see that during the summer observation, atmospheric CO2 concentrations showed a slight decreased trend with the increased height. During the early winter observation, atmospheric CO2 concentrations increased firstly and then decreased with height, with the peak observed at 100 to 500 meters. These two figures show the vertical variations of 30C and 40C. From these two figures, we can see that the vertical distribution of delta 40C and delta 30C, they showed an opposite trend to the CO2 vertical concentrations. The delta 40C and delta 30C tend to increase with height in summer, while in the winter observation, they decreased from near the surface to the 200 meters and then show an increased trend. The ground delta 40C observation showed that the values are generally low. The background values, these low values indicated the fossil fuel CO2 emissions. We calculated the fossil fuel emission input at this site. The excess atmospheric CO2 minus the background CO was mainly from fossil fuel emissions. It accounts about 67%. And the fossil fuel CO2 concentrations were highest in winter and low in summer, We also observed negatively biological concentrations. These negative values indicated the uptake of CO by vegetation in Qingling Mountains. Combine the DAT46 vertical profile, we calculated the contribution from fossil fuel CO2 and from the biological CO2. We showed these two figures. One is summer and the other is winter. We can see that the fossil fuel CO2 showed an increased rate and then gradually decreased with the height in the early winter observation, which indicated the fossil fuel CO2 is mainly from the horizontal transportation. For the biological CO2, it decreased with height during the summer and winter, also in the winter observation, which indicated that biological CO2 is mainly from vehicle diffusion. Combine the 30 vertical profile, we further quantified the fossil fuel CO2 from co-combustion and from the vehicle emission. We found that the fossil fuel CO2 from combustion and the fossil fuels from vehicle also showed an increased and then decreased trade with height during the early winter observation. We further identified the source of biological CO2 and fossil fuel CO2. We observed there is significant positive correlations between biological CO and dirt CO. This correlation showed that biomass burning was an important contributor to biological CO in winter. We observed DAT40C fossil fuel CO local in winter observation, which used this value, we calculated the combustion contribution and vehicle emission. They are 80% and 20% respectively. The decrease in dirt CO2 fossil fuel CO emissions compared to previous measurement indicated that the combustion efficiency is improved in this studied area. Using the LMS backward Higgs-Blende analysis, we found that atmospheric CO2 at this site was mainly from the emission from the nearby city Xi'an and from the transportation from southern Qingling Mountains. This transportation is also confirmed by the CO and the fossil fuel CO concentration peak observed at 100 and 400 meters. Summary, we found the CO2 at this site is increased with obvious spatial and time variations. We quantified the source of CO2 from different sources, and we found the axis CO was mainly from fossil fuel emissions, and in winter, the fossil fuel CO is mainly from combining, and the biological CO is largely influenced by biomass burning. The contribution of fossil fuel CO2 increased with height and then decreased, while biological CO2 contribution decreased with height. The atmospheric CO2 at this site is mainly from Xi'an and the southern Qingling Mountains, which is also confirmed by the CO2 and fossil fuel CO peaks at 100 and 400 meters. In the future, we will continue to the measurement, and we will from the foot to the summit of Qingling Mountains. We'll continue to measure the CO and its multiple isotopes, including 30C, 40C, 80 oxygen, and 17 oxygen, with the purpose to classify the level and to quantify the source of atmospheric CO2 at this site, in this mountain. That's all. Thank you for your attention. La li, can ta li, Thank you.
Thank you for the presentation and the song. Are there any questions from the audience? There is one here. Yeah.
Thank you for this very nice talk and for the great results which you have shown. I wanted to ask about the 13 ratio of coal and the vehicle fleet which you used, I expect, to determine between the coal and the fossil share. So do you know what these two end members have in delta C13?
You mean I separated the fossil fuel CO2 into coal burning and vehicle emission? Yeah. Yeah. I just use the 30C to quantify the fossil fuel emissions into coal burning and vehicle emission. Thank you, Hammer.
The values.
And that's the 13 value of your vehicle. And I was interested how deep.
Yes, we need to know the data 13 of each source. Yeah, we need to know the data 43 for coal combustion. It's about negatively 24%. For the vehicle emission, it's about negatively 28 per million.
Okay. Thanks, that is what I wanted to know.
Thank you.
Thank you. I saw another questions, of course, from the other side.
Thank you for this very nice overview. Are your values representative what you find in China in general for the CO2 concentration? The only comparison I have is the Keeling curve from Hawaii, and there the CO2 concentration is around 420. So it seems what you report is at least 20 to 30 ppm higher. Is that now a general trend in all of China, or is it very specific to your investigation site? Do you understand what I'm asking?
Can you repeat it?
Okay. The most known carbon CO2 measurements are known from the Keeling curve in Hawaii. Yeah, in Hawaii. This is a global measurement of course, a non-urban area, and this is 420 to 425 average value. In summertime, it's actually a little lower, but your values are at least 20 to 30 ppm higher.
Yeah.
And are these values for the larger China representative, or is it different in other areas compared to what you had investigated?
My observation just represents the local emission. It can't represent the whole China.
Mm-hmm.
There are also other sites to measure the CO2 in China. My site just represent the CO2 concentrations at the north face of Qingling Mountains. My site is about 25 higher than the CO2 in the NOAA, in the Monarana, 25 ppm higher.
Okay.
This higher is influenced by fossil fuel emissions.
Yes.
Yeah. Thank you, Bert.
Thank you.
Okay, thank you. Okay, one more hand raised. Let's allow.
Just a question. Is it the atmospheric circulation in that area affect the CO2 level and the carbon-14 as well? Is it the value you show there is maybe bring somewhere else there, or because of atmospheric circulation? Do you have any idea of the air movement in your study site?
And winds.
Okay.
Can you repeat? Sorry.
Okay. Okay. If you don't have any or minimal air circulation, everything you show is there, right? But if you have a strong air circulation, you may bring, that CO2 or fossil or biomass from where else to your study site. This is my question.
Maybe does you mean that the fossil fuel CO2 is mainly horizontal transportation, but biological CO2 is mainly diffusion, vertical diffusion.
Okay, we have discussion later.
Yeah,
okay.
yeah, maybe
We can
move
discuss
this
later.
and
Thank you.
Maybe. Okay, thank you again. And yeah, let me introduce Philippe, I think, Sifoy. Yes, Sifoy from Laboratoire. Now we have the French name of the hydrological laboratory, and he's going to give us an insight at this interface, what's going on between the water and the atmosphere. So, Philippe, stage is yours.
Thank you. So I'm going to present a study about modeling radiocarbon exchanges at the water interface at the water atmosphere interface downstream of nuclear power plants. So first, the context of this study. Nuclear power plants routinely and legally release significant amounts of radiocarbons into rivers. And if you compare the different radionuclides released by nuclear power plants and the different exposure pathways of the population, it was calculated that radiocarbon through ingestion of fish is the most important contributor to the dose to the public, even if total doses remain very low. So it's relevant to study the releases of radiocarbon from nuclear power plants. For that, what we try to do is to simulate the different chemical forms of radiocarbon in the river receiving radioactive effluents. For example, the distribution of radiocarbon between inorganic carbon, particulate organic carbon, dissolved organic carbon, and the different transfers between all these chemical species. This is a picture summarizing the different transfers we try to simulate. And today I will focus my presentation on one specific transfer. That means the diffusion of radiocarbon between water and the atmosphere just downstream of nuclear power plants. If you look at the literature on this topic, there are two different approaches for simulating this process. And these two different approaches are quite contradictory. So this is a copy of a national report. Sorry, it's written in French. But in this report, the authors try to simulate the exchanges of radiocarbon between water and the atmosphere on the Loire River. And I just here underline their conclusions. They concluded that radiocarbon released by nuclear power plants is lost during the transfer in the Loire River, and at the rate of 40%. So 40% of radiocarbon released by nuclear power plants is lost to the atmosphere. If you look at another publication here, published in Radio Protection, the authors studied the transfer of radiocarbon from seawater to the atmosphere in the Channel. And they concluded, it's underlined here, that about 3% of the liquid releases from the LAG nuclear reprocessing plant is lost to the atmosphere. So we have estimation from 3% to 40%. So it's quite different, and it's relevant to study this process. Actually, these differences can be explained by two modeling approaches that are used by the different authors. If we look at the second paper on the Channel River, the authors used an approach where radiocarbon is assumed to follow stable carbon. And so from this assumption, they derived a mathematical model where the next flux of radiocarbon is calculated from the next flux of carbon, according to this equation. And so the next flux of radiocarbon, according to this assumption, can be from water to the atmosphere or from the atmosphere to water, according to season and hour, according to the gradient of stable carbon between water and the atmosphere. If we look at the second paper, they use a different approach where they assume that radiocarbon diffusion is independent of stable carbon gradient. So diffusion depends only of radiocarbon gradient. This is the approach recommended in a paper published in Journal of Environmental Radioactivity. And in this case, downstream of nuclear power plant discharges where there is an over-concentration of radiocarbon in water because of releases, the next flux of radiocarbon is always from water to the atmosphere. So you see a difference between these two modeling approaches. These two modeling approaches are macroscopic approaches, actually, because they calculate net flux of carbon, but they hide microscopic processes. And that's why we tested an alternative microscopic approach based on the random walk theory. This is summarized in this picture here. Here we consider one single molecule of radiocarbon, and it's assumed that this molecule will randomly make jumps up or down with a distance delta during a time step tau. And so you can see a possible trajectory of this single molecule. Now, if you look at a set of molecules, some molecules will randomly go up and some other molecules will go randomly down. And during times, there will be a distribution of molecules in water and the atmosphere. And so the question here is to know what is the probability for a given molecule or for a set of molecules to cross the air-water interface. So this is translated in mathematical forms here. You try to calculate the probability of an event, and the event is the event the molecule crosses the air-water interface. So this, on a mathematical point of view, this problem can be translated because you have to calculate the probability to make car jumps up during n time steps. So this can be simulated by a binomial distribution because this event has two occurrences only, or you go up or you go down, and you calculate the number of up jumps you can make to cross the air-water interface. So it is a binomial distribution. And when you look at a high number of molecules, the binomial distribution can be approached by a normal distribution. and you have to calculate this normal distribution and to integrate it over all the different depths of the river. So now the next problem is to relate the macroscopic theories, diffusion theories, and the microscopic random walk theory. And for that, you look what happens at the air-water interface during the, in this layer where there is a gradient of concentration between water and the atmosphere. And it can be shown, I can not demonstrate this now, but it can be shown that there is a relationship between the macroscopic mass transfer coefficient K and the microscopic coefficients delta and tau. So from this background, we established a strategy for modeling the transfer of radiocarbon from water to the atmosphere. We first observed the fate of stable carbon, and from the fate of stable carbon, we calculate the macroscopic coefficient K. And from that, we can determine the values of the microscopic constants delta and tau. And knowing these microscopic constants, we can apply the random walk model for radiocarbon. This model was tested according to experimental data. So we conducted some years ago 3 sampling campaigns on the Roi River. You can see that on the Roi River there are 5 nuclear power plants in yellow, and we sampled air and water downstream of nuclear power plants during radioactive discharges. So for that, we used a floating chamber. So we measured stable carbon and radioactive carbon in water and air before and at the end of the sampling time in the floating chamber. And from so it's detailed in the publication. And from that, we can calculate the transfer from water to the atmosphere or from the atmosphere to water for both stable carbon and radiocarbon. So this is the results we obtained for stable carbon, where I show here in blue the partial pressure of CO2 in water, and in red partial pressure of CO2 in air. And from this data, you can calculate the global transfer of stable carbon from water to the atmosphere or from the atmosphere to water. It depends on the direction of the gradient. And from that, you can estimate for each sampling campaign macroscopic transfer coefficients K. So from this campaign, from different hours and different sampling campaigns, here I show all the results we obtained. We obtained mass transfer coefficients from 2 to 55 centimeters per hour, which gives the diffusion velocity from water to the atmosphere for stable carbon. And we use this data to derive microscopic coefficients for the random walk model. So these are the results we obtained at the end of the random walk model application. In blue, you see the radiocarbon activity in air at the beginning of the experience, so before in the floating chamber at time 0. In red or orange, you see the measured radiocarbon activity in the floating chamber at the end of the sampling campaign. And in gray, you see the calculated final radiocarbon activity in floating chamber according to the procedure I have described now. So you see that in general, radiocarbon transfer from water to the atmosphere is correctly simulated by the random walk model. But obviously, further experiments should be undertaken because in some situations we don't see enough radioactive releases to estimate an increase of radiocarbon activity in water. So what is the work in progress now? We try to integrate this random walk model into the global radiocarbon model for simulating the fate of radiocarbon in water, and to estimate the quantity of the proportion of radiocarbon which is lost from water to the atmosphere at watershed scale. Thank you for your attention.
Thank you. And the song? You don't participate in the contest.
The song. Oh partigiano, oh bella ciao, oh bella ciao, oh bella ciao, 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 ciao.
Grazie. Okay, now anybody? I cannot see any questions to this modeling. Oh yes, there's one.
Thank you. Thank you for your talk. With all this global warming, how do you expect to change this transfer of CO2, the carbon-14?
Sorry.
With this, the global warming, we expect, I don't know if I am wrong, but we expect to have more water vapor, and how the interface is going to be affected by the global warming and the transfer of carbon-13 in this interface water atmosphere.
Actually, we work just downstream of nuclear power plants, and during radioactive releases. So the quantity, the activity in water during this period of radioactive releases is quite high compared to the atmospheric activity. And so global warming is, I would say, quite negligible for these specific situations, because we are quite close to the release point and not far.
Your talk concentrated on carbon dioxide, radiocarbon dioxide, which is primarily made by boiling water reactors, as I understand. Your talk centered on radiocarbon dioxide, which is primarily made by boiling water reactors. What are you doing similar studies for radiomethane from pressurized water reactors? For radiomethane, 14CH4 from pressurized water reactors, PWRs?
About the methane, do you plan to investigate the methane
Methane
also?
emissions.
The metals.
C14 methane from PWRs as opposed to C14 dioxide from BWRs.
Le méthane? Ah, sorry. No, we don't investigate methane. No, sorry.
We have one more question.
Thanks for that interesting work. Can you very roughly estimate the percentage of this additional radiocarbon emission to the atmosphere, which you just described from the river, compared to the direct radiocarbon emissions from the nuclear power plant? Is it 10%? Is it 1%? Is it half a promille?
Is it low? Is it low? But what, compared to direct atmospheric releases, it's low. We are interested in this part because ingestion of fish is estimated as the most contributor to the dose to the population. So it's important for the river system, but it's not important compared to atmospheric releases.
Okay, thank you. Okay, so let's thank Philippe again. And now, as I see, we stay in France and we move to Paris. And after a short break, we come back to the question of fossil fuel in carbon dioxide, but now it will be also on some other species. Thank you. So, Ingrid.
Yes, thank you very much for the introduction. So I'm Ingrid Chanka. I just started in January this year as a postdoctoral researcher at the Lecce CEA in France. And I want to take this opportunity actually to thank all the people involved in the team that really did a great job, especially with the sampling of last year when I was not there. And that's how I have some preliminary results to discuss with you today. So I'm going to talk about the estimation of fossil fuel fraction of CO2 measured in Paris based on radiocarbon and co-emitted species. And I want to figure out how I can... Oh, okay, it was easy. So I'm working actually in the ICOS Cities project, also known as Pilot Application in Urban Landscapes, so POL project. So this morning we heard a bit about the ICOS network introduced by Sam. So ICOS Cities, it's a bit similar in a way, but focus on cities or urban environments. And just a short overview. We know that, it's estimated that about 70% of the energy-related CO2 emissions come from urban areas or cities, and it's also projected that the urban areas are going to develop or triple by 2050. So in order to tackle with this increase in use of resources and emissions under the current economic system, of course, there is the need for data, models, and services that are aligned together with the policymakers. And this is one of the objectives of this project. So basically, ICOCities brings together and evaluates this innovative measurement approach for greenhouse gas emissions, especially in densely populated urban areas, with this high collaboration with and co-design with city politics. And there are 3 pilot cities being studied in the framework of the project: Paris, Zurich, and Munich, representing 3 different sizes, so small, medium, and large scale. And I'm focused on the Paris region, which is the large scale. And this is an overview of the Observatory of Paris that's composed by several types of measurements and instruments in tall towers, but also mid-cost sensors in roof level. Several things are measured there. But here I want to actually call your attention for these windrows. Sorry, I don't know how it works. The pointer? Okay. So I think you can see my mouse here. Maybe here. Grazie. Okay, perfect. So just to show you these windrows, which shows basically the dominant wind direction over Paris region, which is from southwest to northeast. And you're going to understand soon why that's important. So I'm working mostly with these 2 sites. They are in the greater Paris region. So one site is called Meudon site, and the other one is Romainville. They are about 20 kilometers apart, and they are aligned with this dominant wind direction, southwest, northeast. Here, hope it works. Okay, just to give you overviews. So this is a telecommunication tower in the Meudon forest, and we have different levels here, and there are several equipments installed for this monitoring. So there is meteorological sensors, CO2, CO, CH4, radon, NOx, BC analyzers, and our flask sampler. It's also at inside this room at 65 meters, but air is actually pumped from 3 different heights for the Picaro analyzers. One at 90 meters and 2 others at 65 and 45 meters, and the flask sampler is connected to the 90 meters above ground level. Similarly, we have the other tower, the Romainville tower. You can see it's more urban environment. It's after the main Paris. It's also a telecommunication tower, and it's also a super site, so with every instrument, meteorological, NOx, BC, and greenhouse gases, radon, another flask sampler, and the multi-component gas analyzers. And here, the flask sampler is connected at 103 meters above ground level. And about the sampling strategy, so then basically we follow a Lagrangian approach. So, in simple terms, I receive triggers by mail every 3 hours, and they're going to, it's basically back trajectory based on the German, how is that? Deutsche Wetterdienst, so German weather service, where basically, these back trajectories are calculated, and there is an estimate of when the air masses are going to be following this dominant wind direction within 5 kilometers from the towers. And I just did this video this morning at 3 a.m. It's exactly the air crossing over Paris, because I received this trigger there. But we also received triggers in the other direction, and of course they are much less frequent, but we also have this possibility of sampling at this direction. And receiving the trigger, of course, is not enough to decide to keep the sample, because we need a confirmation. So when we receive the trigger again, or the email again after the air mass has supposedly passed, then we consider that a confirmation, and we keep the samples. So samples are always collected in pairs. And just to show, oops, sorry, let's try again. Okay, just want to show you, because the other one was really aligned the sites, but sometimes you have in the 5 kilometers, but always the target site, it's at 0 kilometers. And we have then samplers in both sides. So here at Meudon, we have a sampler, we call it a Ringo sampler. It was built by the University of Heidelberg in the framework of another project they had in the past. And it has space for 8 flasks, 3-liter flasks, the standard IQOS flasks. And at Romainville site, we have the IQOS automated flask sampler, where you can have up to 24 flasks. And just overview of the status of sampling. So samples started to be collected in March last year, and they are still being collected now. And so far, I counted 115 pairs, so 230 flasks collected, until today. Not all of them analyzed yet, of course. But and this is in the dominant wind direction. But as I mentioned before, there is the other wind direction where we can sample too. Of course, they are much less frequent. Yeah, we have 14 pairs from this period. So in principle, as an exploratory approach, I'm analyzing them or calculating quantities with them separately. So first, an overview of the comparison between the CO2 mole fractions measured in the flasks. At the flask, we measure several trace gases, besides delta 13C and 14CO2, and compared with the picaros inside. And in general, there is a good agreement if you take the mean value. Of course, there are some large standard deviations. These come especially from the in situ instruments. And you also take the opportunity to remove some outliers, as for example, some strange samples with a 5 ppm difference. And we saw many times already this week these 14C based fossil fuel CO2 estimates. But basically here what I want to call attention, so in the final equation, the mass balance, in this case we have our background site not at Mace Head or another site more close to the ocean, but is actually one of the sites that are aligned in this dominant wind direction. And when we are in the southwest to northeast, then our background station is Midon. And when we are in the northeast to southwest, then our background station is Romainville. And there are, as you know, there are some corrections we have to do for example, nuclear 14C, that especially in France can be problematic. But at this, this alignment of the sites, it's not a big problem. Okay, so some of the results we have so far. So here it's from all the pairs that we have measured until now. And we can see that there is variability throughout the year, especially the winter months. They have a much higher fossil fuel as expected. And here I tried to see aggregate. Sorry, I didn't put January, February, but the data of January and February is also included here in the winter period. It's just the label that is not correct. But basically here I want to see how the aggregated data would look like, especially in these two kind of different origins of the air masses, if they are coming more from the inside or more from the closer to the coastal side. And in general for the fossil fuel, I would say there is not such a huge difference, but we clearly can see a difference in the non-fossil fuel estimates. And this is the non-fossil fuel, which we are aiming to compare with the biospheric simulations of the models afterwards. They are simply the difference between the total CO2 that aggregate the data from the picaros in the same wind direction, minus the 14C based fossil fuel CO2. And let's take a look also at the co-emitters. So here to show you separately by season. For the delta CO, so the enhancement of CO, there is not a really good correlation, especially in summer and autumn, and the best correlation is in winter, where they have a much more fossil fuel signature. And just zooming here, I'm focusing on the dominant wind direction, where we have also much more data. And we get to this RCO of 5.5 that we can try to compare with the inventories. So there is the inventory of Air Paris that covers Paris region, but also the Ile-de-France. And here I'm showing you the one that I had available for now, which is 2019. There is a new release from 2022, but I couldn't yet include it. But anyways, we can already take a look at them. And yeah, if we average the month, so especially in March, there was no difference between the years, but all of them fall always in between what would be the baseline of different sectors. And yeah, 5.5 would be here between roads transport and shipping. And taking a look also at the NOx, so it's very similar in the sense that the correlation in autumn and summer. Sorry, actually, okay, but maybe in the next slide is correct. Yes, I'm so sorry. Okay, so let's focus in the winter, but believe me, it was not good for the summer, the NOx either. And yeah, we get this different factor here, also in the dominant wind direction. And yeah, from the air Paris for the NOx, we have also basically a distribution of what would be the main sectors. So 55% would be actually in the road transport, and followed by 11% in the residential sector. And we kind of see this pattern with our samples too, but not for all the year. So that's something that needs more investigation. And unfortunately, at least at this release, the black carbon is not so, I don't know, I don't want to say it's not so reliable, but yeah, there are some issues. So I think it needs more thinking and proper work. So as a summary and the perspectives of these first 8 months of work from my side at least, but not from the whole team, of course. So direct estimates of fossil fuel CO2, of course, are a big advantage because they are actually isolating this fraction that what the policymakers need for achieving their policy targets. These, what I consider preliminary results, give already some insights, especially in the non-fossil fuel fraction, which is, I think is going to be very important, especially to the level of comparison with models. The uncertainties can still be a challenge, however, especially for the summertime. And yeah, I find especially nice that this approach of sampling according to the wind direction can facilitate the calculations and all the issues with, for example, nuclear contamination that can be really large in the region. And we hope that we can improve the comparisons with the inventories, expanding, of course, for the black carbon even more, and to filter according to the rush times, especially for NOx and BC. That's really important. We sing together. Thank you very much.
Okay, thank you. So, does anybody raise a hand and ask a question? Ingrid. I see no questions. sleeping audience.
We have a whole dinner to discuss all of this. No.
Okay, maybe they are too shy. They will ask you during the evening.
No problem.
Okay, thanks.
Thank you.
So, yeah, thank you. And last but not least, we have the representatives of our hosts now to show us the study on waste and, of course, using the radiocarbon. So the speech will be presented by two persons, and David will start. Yes.
Good afternoon. I am Davide Sanghez. I work in the Addoambiente Laboratory located in Cascina Maggiore, Giussago, near Milan. The title of this presentation is Comparative Study of Waste Analysis Methods for Biogenic Fraction Quantification in Waste-to-Energy Plants. This activity was carried out in collaboration with CEDAD. The objectives of this study are determine the biogenic fraction in waste with selective solution method and accelerator mass spectrometry. Number 2, compare results. Number 3, identify strengths, weakness, and areas of improvement. 176 samples from 8 waste-to-energy plants was analyzed between September 2022 and December 2023. The methods used for determination of biomass content in according to UNI and ISO 21644 are selective solution method carried out by Addoambiente, which is a chemical method, and accelerator mass spectrometry, AMS, instead is a radiocarbon method. The first step of sample preparation involves drying the sample at 105 degrees Celsius. After, the sample is grinded to 1 millimeter for selective solution method. To make even more homogeneous for AMS, the sample is grinded to 0.2 millimeter. I will describe the steps for determination of biomass content expressed in percentage by mass. Weight 5 grams of dried sample, add concentrated acid sulfuric, and stir for 16 hours. After 16 hours, add concentrated hydrogen peroxide and stir for 5 hours. After, add the mineralized water to the sample and then filter. Dry the sample with the filter at 105 degrees and wait. Instead, to determine the ash content, the ISO 21656 is used. How is the result expressed in percentage by mass? There are WB, biomass content expressed as percentage by mass, WNB, non-biomass expressed as a percentage by mass, and A, ash content in percentage by mass. WB plus WNB plus A equal 100%. For determination of biomass content in percentage by calorific value, the calorimetric BAMB is used. The ICA C6000 was used in the Addoambiente laboratory. For determination and expression of the result in percentage by calorific value, it is necessary to determine Q, calorific value of the sample, Q residue, calorific value of the residue, and it is necessary to determine QB, calorific value of the biomass fraction. VB call, biomass content expressed as percentage by calorific value, it is calculated by this formula. For determination of biomass content expressed in percentage by total carbon, the elemental analyzer is used. The Lecce TrueSpec CH chain was used in the A2 Ambiente laboratory. For determination, the expression, the results in percentage by total carbon, it is necessary to determine C tot, total carbon content of the sample, C residue, total carbon content of the non-biomass fraction, and C hash, total carbon content of the hash fraction. The biomass content expressed as percentage by TC is calculated by this formula. Usually, A times CH equal 0. On which principle is the AMS method based? The AMS method is based for the transformation of the sample in carbon dioxide by combustion, and then in graphite with by reduction. The biomass content expressed in percentage by total carbon. This method is used in different fields and applications, such as biofuels or bioderivated plastics, for analysis of waste, or carbon dioxide emission from stationary sources, as power plants, for determining the renewable energy fraction. The process involves grinding the sample to 0.0 millimeter for homogenization, combustion to carbon dioxide at 900 degrees, graphitization with hydrogen and iron, and measurement with accelerator mass spectrometer. Thank you for attention. I give the floor to my colleague Alessandra Panzitta.
Good afternoon, I'm Alessandra and I work for the Waste to Energy Monitoring and Support Department of A2A. And now I'm going to show you the results and the final outcomes of our study. As we said before, our study focuses on the analysis of different types of waste treated in various waste-to-energy plants within the group. The waste analyzed are the MSW, which stands for Municipal Solid Waste, SW, which refers to special waste defined by Italian law as any kind of waste that is not municipal solid waste, and two types of special waste, RDF and SRF, so refuse-derived fuel and solid recovered fuel. The difference between two types of fuel is that SRF is an high refined fuel produced with very stringent specification on moisture composition and calorific value, while RDF in this case is produced by only municipal solid waste and with less stringent specifications. And here I reported the number of analyses conducted during 2022 and 2023 and the type of treated waste for each plant. Here you can see the average of biogenic fraction of waste for the different type of waste-to-energy plant, and the results obtained are consistent with those reported in literature, showing a lower biogenic fraction for pretreated and special waste, as you can see for waste-to-energy plant 7 and 8, with a percentage around 45%. And generally, municipal solid waste has a higher value of biogenic fraction, and it is also confirmed by the two analytical methods that we use, so the selective dissolution method and AMS method. and obtaining a result of around 60%. We also can notice that the two analytical methods are in good agreement. In fact, the results obtained are quite similar. Here I reported the results, but in this case not only the average value, but also the minimum and the maximum value, in order to enhance the variability of results obtained. In fact, this high variability obtained by our results is due to the heterogeneity of waste, especially for the municipal solid waste. In fact, as you can see, for waste-to-energy plant that treated SRF waste, there is a lower variability in results and better repeatability, because this waste is produced with a pretreatment which homogenized the material. Here are reported a number of analyses by differentiating between the type of waste analyzed. So municipal solid waste, special waste, and SRF, which we consider, as I said before, because it is a waste with specific characteristics. And as you can see, the number of analyses for the two analytical methods is almost the same. So the comparison between the two methods can be done directly and is relatable without any doubt. Here, the average results of biogenic fraction of waste for these three types of waste. And as you can see, the results are quite similar, so the two techniques are in good agreement, especially for SRF that I said before, presented the results with a lower variability. So after the analysis of the result, our aim, our objective was to identify strengths and weaknesses of these two analytical methods. Here we reported pros and cons for the first method, so selective dissolution method. We have to say as pros that is a time and cost-effective method. It's possible to treat more sample at time and to obtain rapid results. It's a simple method, and the results can be in different output formats, so on calorific value, on total carbon, and on mass. As cons, we have to say that it is a method with a high uncertainty, and there are problems with materials with high biomass content but poorly biodegradable, and problems with high fossil content but high biodegradable materials. Another problem is the low representativeness of the method, because the sample that is analyzed is a small sample, only 5 grams, and it's not representative of all the waste metrics. Then, talking about the second method, so accelerated mass spectrometry method, an important pro is that this method is high accurate, so the technique is able to count every single atom of carbon-14, and it's also high sensitive. So the analysis, it can analyze both biodegradable and non-biodegradable materials. On cons, we have again the low representativeness of the method. In fact, the sample in this case is also smaller, 10 or 20 milligrams, and it necessitates high specialized equipment, so requires expensive and sophisticated instrument. To summarize our results, the analysis shows values of biomass content consistent with those reported in literature, so high biogenic content for municipal solid waste and lower biogenic content for special waste. Then an high variability in biomass content expressed on carbon content regardless the technique used, and also low variability of SRF results, as you can see in the graph below. The data variability is mainly due to the difficulty in achieving an homogeneous sampling of the load, and also to the low sample quantities analyzed, especially for AMS, as I said before. So in order to use waste analysis to characterize the biogenic content of a waste-to-energy plant, it's advisable to conduct multiple analyses of every waste streams entering the plant over the course of the year, and to calculate then the final average. But at the end, as the final remarks, what we have to say is that given this high variability observed, waste analysis is not the most suitable method for evaluating the biogenic carbon fraction of a waste-to-energy plant, but it's still a useful data. In fact, we can use waste analysis to identify and map the biogenic fraction of the waste input. We can use it to validate and compare that with the emission analysis data. And also we can use it to calculate emission factor of the different waste streams. Then the measurement of carbon-14 of samples taken directly from SAC emission are less variable. And it is due to the homogeneous nature of a flue gas in respect to the heterogeneity of waste. So the sample taken is representative of the emission, resulting from the fuel mix fed. And in this case, AMS can be used to its full potential, providing the most accurate results. Thank you.
Thanks to both of our speakers. Are there any questions from the audience? We are not used to, we are used to seeing the small sample size as an advantage, but in your case, I think it's the opposite.
Yes, it's, in our case, it's the opposite because waste is heterogeneous, so.
Okay, I see Walter.
Is my impression correct that most of these conversions of biogenic waste into energy ends up with 60 to 70% biogenic fraction? And it seems that we heard other reports today, and that it looks like always there's this 70% remaining of biogenic material. Is that true, or?
60% is a good result for us because it's a result that we obtain in plants in which have municipal solid waste. So yes, it's around this number, but in the most cases is around 50%, 50 and 60%, yes, but it's correct.
Okay.
Okay, we have another question.
Thank you. Yes, we did similar work on incineration, waste incineration plants, and it was typically 50%, but the operator said that the sort of waste they combust depends very much on the status of the economy. So if people throw away their furniture or not, that makes a difference. But if you would really want to monitor this, then probably you have to sample integrated CO2 of the flue gases for 24 hours and measure that and do that day after day. Is that in the end the conclusion?
No, I think that it's not necessary to do this every day, but I think that with a monthly frequency, we can characterize the plant, so with the sampling of the flue gas emissions. With waste is more difficult because the results are variable depending on the size of the sample. So I think that's the best choice is to monitoring biogenic fraction by due sampling analysis, sampling and an analysis of the flue gas, and I think that the frequencies, the optimal frequencies, is by monthly.
But then you have to monitor the flue gas day after day, depending on
Yeah,
what
yeah,
they combust. That's what I mean. Yeah.
Yeah, the monitoring, yes, but the analysis. We say that I don't know, but the flue gas is for us is constantly monitored, and we are obliged to monitor indeed. So, but depends on which parameter. CO2 is not an parameter that we have to monitor, but in the end to do this, yes, constantly. Yes.
Okay, thank you. Thanks for your presentations, and let's thank all the speakers for the presentations they have given on these tiring sessions. Yes. Okay. Do we have any announcements from the organizers? No, everything's been said. You remember the time that we meet? Yeah. Everybody, just it's 5:15. Okay.